Hi, everyone. I'm Thea Steinhardt, and I'm the president of the TNT network, and I'm here to welcome you today to the first TNT Showcase. And so this has really been an event that I've personally wanted to see since I joined Johns Hopkins. So I joined knowing that there's a wealth of labs working on neuroengineering projects across all of its campuses. And through this event, for the first time, we have the opportunity to highlight their excellent work and to bring together this large community at Hopkins interested in neuroengineering. So for this very first event, we've actually had 15 groups of presenters at all different stages volunteer to share their projects with you. Because it's the first time, there's likely going to be some kinks and we're likely going to go over time. But the goal here is really to have a good time and share some really interesting ideas. So we're hoping that you can stay to the end to hear everybody's excellent talks. So let's get started with day one. Today is the first day of a two-part event. Where we're going to have 9 different presenters here to share their work with you. For both days, we're going to be joined by our judges, Jean Friedman, who hopefully can wave at everybody, professor of the otolaryngology and BME department, Chuck Montague, the BME Industrial Liaison Associate, and Joshua Vogelstein, an assistant professor in the BME department. They're not only going to be here to bring their expertises while scoring the presentations, but will also be bringing their personalities, I'm sure, to the feedback of all of the presentations right after each talk. Today's event is going to be emceed by 2 of our TNT board members, Gulnush Kamali, if you can see her, and Alex Weiss. And Gulnush will be introducing each of our presenters and starting the clock. And the presenters will either have 5 or 10 minutes to share their work with us. At the 30-second mark, Alex is going to play this bell sound. So hopefully you're going to be able to hear that. And then exactly at the 5 or 10 minute mark, we're going to have an alarm go off and we're going to cut the presentations. And then audience members, please stay to the end of today to vote for your audience favorites out of all of our 9 speakers. On day 2, which is going to be on April 13th from 3 to 4 p.m., they're going to have a second set of presenters. So stay to the end of day 2 as well to vote for your overall audience favorite. And remember, audience members, you're not only involved in judging too, but you're also capable of winning prizes for asking different questions and giving feedback to the presenters. When the presentation's going on, if you're having an idea or a question, write the name of the presenter into the chat box, and then Alex will call on you at the end of each presentation. So with that, I'm going to hand the event over to Gulnush and Alex, and we're really excited to get started.
Thanks, Thea. Thank you everyone for showing up today. We're very excited. And I don't want to take up too much time since we are on a tight schedule. So I will just say your name, present you guys, and you can pull up your PowerPoint. I also want to apologize if I mispronounce your name. I will try my hardest to pronounce it correctly, but I apologize if it is incorrect. So our first set of speakers, it's a duo. It'll be Tarana Parvez Cavosia, I'm really sorry, and Molly Accord, and they're part of Dr. Amir Mambachi's lab. Whenever you guys are ready.
Hi, can you see our screen? Yes, okay.
Yes.
Hi, my name is Molly Akard and my colleague is Tarana Kovasia. We're master's students in the biomedical engineering department. And today we're going to be sharing with you guys sonodynamic therapy in the treatment of glioblastomas. GBM is the most common malignant primary brain tumor. It's aggressive, invasive, and infiltrating in nature, which means its victims have the shortest survival rate. The average survival is 12 to 15 months, and only about 11% of patients survive to 5 years. Due to the microstructures GBM develops, total resection is typically impossible, leading to a high recurrence rate. During the process of focus ultrasound, acoustic waves are generated by a transducer and are designed to converge at a focal point, which can be at a tumor's location. This focal point constitutes maximum acoustic energy, which can be used as a host of therapeutic applications, for example, thermal ablation, blood-brain barrier opening, and sonodynamic therapy. In sonodynamic therapy, a sonosensitizer, 5-ALA is what's used for GBMs, is injected intravenously or administered orally and is preferentially taken up by malignant cells. Within these cells, the sonosensitizer undergoes metabolic processes. Focus ultrasound can be applied to the product of said processes. 5-ALA's product is fluorescent PPIX. This generates reactive oxygen species, or ROS, which is then cytotoxic to cancer cells.
Sorry. Since this is a relatively new form of therapy, there isn't a lot of literature out there, but the work by Dr. Hinenen's group at the University of Toronto is very promising. In their study, they demonstrate the efficacy of sonodynamic therapy to treat C6 glioma model in rats. In figure A, you can see the treatment plan in a representative rat. To treat a larger volume of the tumor, a 16-point sonication plan was selected. In figure B, a representative image showed the coronal and axial planes of the tumor on day 28. In the center of the tumor, a non-enhancing lesion was found, and this corresponded with the area that had been sonicated. The surrounding enhancement corresponded to a tumor growth, since the treatment didn't get the entire tumor, but this study was successful in demonstrating the efficacy of sonodynamic therapy. We want to build on that, which is why in our study, we're looking at the U251 human glioblastoma cell line, both in vitro and in vivo. And we're mainly looking to optimize treatment parameters. That is, how many repetitions of the same treatment would be required to hopefully get the entire tumor over time. And that brings us to the RK300, which is an MR-guided focused ultrasound system for small animal studies. It can be used to perform several focused ultrasound applications like blood-brain barrier opening, ablation, neuromodulation, and in our case, sonodynamic therapy. The RK300, including the one at Hopkins, it can be broken down into two parts, the front end and the back end of the system. The front end is what we call the RK300 arm, which consists of the transducer and the positioning system that mechanically steers the focus by physically moving the transducer. The back end consists of the driving electronics. That's your power source, your function generator, your RF power amplifier. The arm of the RK300 that goes into the board of the magnet, while the driving electronics, they stay outside the MRI room. And a governing software user interface can be used to plan and carry out treatment, while magnetic resonance imaging can be used to guide treatment and observe therapeutic effects, if any. And we will be using this system for our in vivo studies, and we're really excited to see the results they produce. And that's about it. We hope you enjoyed our talk, and we would love feedback and questions. Thank you so much.
Thank you for that great talk.
And you guys exactly 4 minutes.
Woohoo! That's awesome. So
Wow.
now we'll open the floor to questions. Like Alex had put in the chat, if you can put just like Q, and I guess now we don't have to put the presenters' names because they're here. Or if you'd like to unmute yourself and ask a question, please do so. And if not, we'll also look to the judges for whatever feedback they may have or questions for the presenters.
I have a question.
Oh, yes, go ahead, Dr. Freeman.
So, first of all, what frequencies are we talking about for this ultrasound?
So we're talking about a center frequency of the transducer of about 1.06 megahertz. So because we don't need a very low frequency, because we don't need to go too deep because it's a small animal. And so we need a smaller focus because again, it's a small animal. So 1.06 megahertz.
How many transducers do you actually have?
It's a single transducer, and if you want like multiple focal points, it goes serially. It goes between each of the focuses that you select. Like in that 16-point plan, it goes one after the other. So it sonicates one point, it moves to the next, and that mechanically the transducer is mechanically moved.
Oh, and I guess the idea is that the reason why it kills those cells specifically is because those cells vibrate more than other cells.
No,
yeah, go ahead, Molly. Sorry.
So it kills those cells because so the interaction between the focused ultrasound and the PPIX that's generated from the 5-ALA, that's what creates the ROS. So hopefully, I mean, it's been shown that 5-ALA is preferential to GBM cells. So 5-ALA should only be within those cells. So even if you were to apply focused ultrasound to healthy cells nearby, they should not have the same cytotoxic effect because they should not contain 5-ALA.
Got it. Thank you.
Mm-hmm. Great. Thank you guys so much. Round of applause for our speakers, and we'll have to keep it moving. Sorry, I'm not trying to like be rude or cut it off, but we got a lot to get through. All right, so our next set of speakers is Eva Yezerets from Dr. Adam Charles' lab. Eva, if you have your stuff ready.
Hi, yes, just a second. All right. Can you guys see my screen?
Yes.
Yes. Okay, thank you. Okay, good morning. My name is Eva Yezeretz, and I'm a first-year PhD student rotating with Professor Adam Charles in biomedical engineering. Today, I'm going to talk about how we can use manifolds to represent neural states and why we can benefit from alternative dimensionality reduction methods as part of that. In Dr. Charles' lab, Neural Signals, we take a systems neuroscience approach to understanding how neural circuits interact. So we extract and interpret structure and function from neural activity data, such as calcium imaging. This is an example video of a rat navigating a virtual reality maze. And on the right, you can see the fluorescence of different place cells in the CA1 region of the hippocampus as the rat explores. The data that I'm working with for my rotation project is from our collaborators in Professor David Tank's lab at Princeton, and our pipeline is a modified version of a manifold learning method from a work in progress paper by Ryan Lowe. From their experiments, we get to work with both large-scale place cell population recordings from the dorsal CA1 region, as well as behavioral data. So when we look at this neural activity, we can ask ourselves, how do we represent this data in a lower dimensional geometric representation that preserves relationships between relevant states? Do we see recognizable patterns? For example, in a spatial navigation task, are there orthogonal dimensions accounting for X and Y location on a map? Is there other interesting cognitive activity going on, and how well are we capturing it, and what does it all mean? So the analysis tool that we are using is manifold learning. Manifold is a representation, a geometric representation that preserves complex nonlinear relationships in the data. So here's the classic Swiss roll example. The difficulty is that manifold methods don't scale well, and our data has dozens or hundreds of dimensions, one for each neuron. So we need dimensionality reduction as an intermediate step to reduce computational demands, and also to make the manifold that we extract easier to tie back to behavior. Principal component analysis is a classic dimensionality reduction method that many of you know. It captures orthogonal dimensions ranked from greatest to least variance explained, but it filters out the high frequency, low variance dimensions. And with neural recordings, we suspect that those high frequency dimensions may actually represent more complex cognitive states, so we don't want to lose them before learning the manifold. So we're testing a method based on the principles of compressed sensing to perform dimensionality reduction in a way that still captures the signal while not low pass filtering the data prematurely. So given some data X, so here we have D dimensions by n equals 1 samples, PCA reduces dimensionality via phi, which is the high variance subspace. So instead, we propose a method based on random projections where phi is a matrix of random values with dimensionality k by d. So as long as k is smaller than d, but large enough, theory, for example, the Johnson-Lindenstrauss lemma, proves that the manifold geometry is preserved in the low dimensional projections. So here, we've done some initial experimentation. Here we've compared our method, so it's shown as RF in orange, to PCA, where we have a very simple set of signals. So each neuron is represented as a cosine function, and they all have different frequencies. In this DFT plot, the vertical lines are the ground truth frequencies. And you can see that the max of the first 3 dimensions extracted by the pipeline with PCA, so that's shown in blue, captures fewer peaks than the pipeline with our new method, shown in orange. So we've also done some comparison to a simulated 2D random foraging task. Both methods capture, first of all, 2 orthogonal dimensions, so we think those correlate to X and Y in a space, but it's a bit tricky to show this with just this one set of figures, but what we've seen in general is that PCA seems to extract a bowl shape for this additional dimension 3, and our new method starts to extract more of a saddle shape. Again, not super clear from these figures, but this might be evidence of additional or more complex cognitive network states rather than just place cells firing based on XY location in the environment. But we're doing further experimentation to hash this out into a clearer story. So looking forward, we're going to test on more low dimensional examples such as the ones I've described, as well as real rat behavior and calcium imaging. And our compressed sensing method should be faster and less computationally intensive, and we should be able to run it in real time, especially since videos can be directly processed via Fourier transform. So
that's 5 minutes.
Okay. So,
wonderful.
Yeah, thanks very much.
Thank you, Eva.
Really
I think
good.
we have a question from the audience. Ehsan asks, Ehsan, would you like to ask your question yourself?
Sure. So it's just very quick. So you just compared your technique with PCA. How about the nonlinear dimension introduction like TSNE or specifically UMAP that are also nonlinear and can preserve the manifold thing?
Yeah, thank you for that question. So the paper, the work in progress paper that we're working with has done some comparisons with other methods. I think they compared to LLE or local linear embedding. I haven't done comparisons to TSNE or UMAP yet, although that's something that we're considering. So far we're trying this sort of more signal processing inspired method because of the frequency content of the signals, but that's definitely part of the additional experiments that we need to do is compare to other dimensionality reduction and embedding procedures.
Do we have any other questions from the audience? If not, maybe we could get one of the judges' perspectives. Dr. Vogelstein, do you have any feedback or questions for Eva?
Sure, Eva, can you go back to the slide where you explain your method?
Yes, yeah. This one? Or
yeah, so is your method literally random projections?
It's not just random projections. I can't, so the trouble is that the work isn't published yet that we're working with to inspire the new component of the pipeline. There's some FFTs in there, and I don't know how much I'm allowed to talk about, but this is part of it. This is a major change from PC.
I see. Okay, thanks.
Okay, yeah, thank you.
Are there any other questions? Any feedback, thoughts? No? Yes, we can move on. Next one. One thing I want to mention is please feel free to turn on your video and clap for our presenters, or at least like do the emoji clap sign as well. Like, it's, make sure we all know that everyone is trying really hard, and we're really happy to have them here. Our next presenter that we have up is Soon Hoon Lee from Dr. Gislin Diagnozab. I'm so sorry. I'm very bad at pronouncing. Even though my name is difficult to pronounce, I have even a harder time pronouncing other people's names. So I really apologize.
Yeah.
I'm so sorry for screen sharing. I got the order wrong. My bad. I'm so sorry about that.
No, yeah. So my name is Sihan, and it's Dr. Jalan Danieli.
Thank you very much. Sorry about that. I apologize.
No problem. Can you see my screen?
Yes.
Okay. So hi, everyone. I am Suhyun, and I'm a research assistant in Dr. Danieli's lab. I'll be talking about phosphine mapping strategies for blind recipients of the intracortical visual prosthesis. So let me start with a brief introduction about the implants that we're developing. The goal is to generate functionally useful visual perception by sending direct electrical stimulation to the visual cortex. This stimulation produces small spots of light, which we call phosphenes. And these phosphenes resemble pixels in a computer display. And the idea is that they can easily be combined to generate coherent images. Even though our lab is already successfully working with blind subjects with retinal implants called Argus-2, we want to explore intracortical implants because many blind people with acquired blindness have damaged eyes or optic nerves. Because retinal implants require the subject's eyes and optic nerves to be intact, many blind people would not be eligible to receive retinal implants. By developing intracortical implants, we can bypass the eyes and the optic nerve by directly transmitting visual information from the camera into the visual cortex. Intracortical implants are more complex than retinal implants because the area we want to stimulate is in the folds of the calcarine fissure. However, we are hopeful that these implants would benefit a wider range of blind people and because we have a larger area of the brain to work with, we hope to achieve higher resolutions of vision for the blind subjects. So here's an example of the kind of vision that can be created using these implants. Oh, it's lagging, right? Can you see any of the changes that are happening?
No.
Yeah, so basically he's seeing different like shades of light through these phosphines. Yeah. Okay. So before we test such implants in human volunteers, we want to estimate the performance of these implants by running psychophysical experiments on normally sighted participants. By simulating phosphines in healthy subjects, we can develop reliable methods to map them out in blind people with implants. In our psychophysical experiments with healthy subjects, we ask the participants to wear a virtual reality headset and a finger tracker sensor on their index finger. The participants will see a green dot in the middle of their screen, and we provide them with this green dot to give them a reference point to the center of the screen. They will also see white dots of varying sizes in the periphery throughout the experiment. These white dots represent phosphines that we hope to generate in blind subjects by stimulating the electrodes. We ask the healthy subjects to point at the dots and also ask them to look at the dots either by moving their eyes or moving their head. We collect data on their head and eye orientation, finger location, and display dot location throughout the entire experiment. We use these data to create a map between the dots they see in their screen and the perceived location of the dots calculated by the finger, head, and eye data. So in conclusion, we're in the early stages of developing these intracortical implants. There are many other features that need exploring, such as the most effective amplitude of electrodes, or the frequency of stimulation, or the pattern of stimulation. Currently, we are collecting data from the psychophysical experiment that I just described to estimate the perceptual effects of cortical implants. Even though these maps do not consider the surgical limitations of the irregular mapping of the visual cortex, we can use the maps created with healthy subjects to estimate the visual maps of blind subjects with intracortical implants. More realistic phosphine maps are expected to be obtained when we start preliminary placement of electrodes in early volunteers that we hope to have in a few months' time. Thank you.
Thank you for that. It was a really interesting talk. Are there any questions from the audience? Also, just be aware, if there's no question from the audience, it doesn't mean anything. This means you did such a great job on your talk that there's like, no one needs to get any clarifications. If not, Dr. Montague, do you have any suggestions or feedback or questions?
Yeah, I thought it was a good presentation, really clear. And I think it's really clever that you're using the visual, the normal sighted people to map that. But I guess I didn't understand how mapping where you track their head motion and eye motion can be used then to map back into the mapping that needs to be done with blind people.
Right, so we would have to make the connection once the implants are in the earlier volunteers to see which electrodes would map out to which location in their visual field.
So you have to, do you have to do the same tests with the blind patients? Is that what you're saying?
We will be doing this with the blind subjects once they have the implants.
So it really is just learning the methodology. It's not, you're not going to be developing a technique with the visually sighted patients to be able to use that. Okay. Oh, no, it's a clever test.
And I think we had a question from the audience as well. Alex, would you like to ask it?
Sure. Do you anticipate differences in mapping locations in blind from birth individuals compared to blind after birth individuals?
I think we would anticipate there to be some differences, and we expect individuals to have different types of mapping, so we would have to thoroughly test this out.
Right. Are there any other questions, or are we out of time?
Yeah, I can ask a question. So I don't know if your lab has that, but maybe stimulating even in a healthy individual with a electromagnetic pulse or magnetic pulse, just to have a stimulation in your mapping that might help as well. Have you considered that, or do you have any plan for that?
So we don't really work with that, but that sounds like a good idea.
Alex, how are we doing on time?
She's been perfectly on time. We are about 8 minutes behind the overall schedule, but
Okay.
I think we're doing okay otherwise.
All right, well then in that case, I know there was another question in the chat, but I think we might have to move on. If you wanna, we can give everyone's information afterwards if you wanna contact the presenters and ask for questions that way. So our next presenter is Adriana Iotos. She's with Dr. Charles Della Santina's lab. And whenever you are ready.
For sure it's me this time, right? I know I got a little excited last time. Okay.
And we love the enthusiasm.
Just making sure. I was going off the schedule. That was my bad.
No, you're good.
Sorry, I will start it in just a second. Okay, there we go. Perfect. All right, and I've got my own timer as well. Hi, everyone. My name is Adriana. I'm going to talk to you a little bit about the human clinical implant trial that we have for a vestibular implant, and I'm going to get right to it because 5 minutes is short. So what does the vestibular system do? Balance, generally, but specifically, I want to talk to you about how it drives stabilizing reflexes. Okay, anyone who was out this winter and stepped on some ice has definitely needed your help of your vestibular system to stay upright. And actually right now you can test some of your eye stabilizing reflexes if you just look at me or my slide. Actually look at my slide, look at some words and nod. Just nod pretty vigorously. You can still read that. How can you read that? Your vision is stable because your eyes are moving equal and opposite to your head. Okay, so if you have vestibular hypofunction, your vestibular system isn't performing as normal. You might have abnormal gait. Oscillopsia, which is the word that describes blurry vision during head motion because you don't have that eye reflex that you need, and lower quality of life. How can we restore function to people with damaged vestibular systems? Well, there's no current therapies that fully work. And that's why our group in 2019 published some data describing how we can partially restore some of the eye stabilizing reflexes using a vestibular implant that people continuously use, meaning we give them the device and they walk away with it and they come back a couple months later and we continue to test and look for the efficacy. Briefly, how does this device work? Well, let's see, there we go. Hopefully you can see my mouse. The inner ear of the vestibular system, the part that encodes for head rotation, basically looks at 3 different directions. That's pretty good, right? We like things in 3D. So here's my right-hand rule. Hopefully not getting flashbacks to physics. Okay, so we innervate the 3 branches of the vestibular nerve that innervate these 3 different directions. And then using a mapping that we set actually individually for each subject, we stimulate with a different pulse frequency and a different pulse amplitude depending on the head rotation that we detect from the gyroscopes that are situated on the subject's head, on this head-worn unit. So it's magnetically coupled to their head. And when they move, the pulse frequency and pulse amplitude will go up if it's in the direction of the implant, and it'll go down if it's in the other direction. So it gives them some information about their head motion. Right now we've got 9 subjects, started in September of 2016. Most recent person was implanted, subject 9, in August of 2020. Most recently, we published something earlier this year with the posture, gait, and quality of life and hearing outcomes for the first 8 subjects. Very briefly, for posture, right here on the left, 2 out of 2 items, or 2 out of 2 metrics were better at 6 months and 1 year with the device on for the subjects. And for gait, 2 out of 3 of the metrics were better. Obviously, much more in detail in the paper. This is a quick 5-minute talk. It's a quick overview. And for quality of life, 2 out of 2 metrics for dizziness and disability were better at 6 months and 1 year, and 1 of the 2 metrics for quality of life was better at 6 months and 1 year. But here's what we're actually doing right now. Perfect. I have about a minute and a half to tell you. So an interesting new project we're working on. So head impulse tests. I did some fun testing with you. We're going to do some more testing. So look at me again. Actually, look at me because I'm going to demonstrate the motion. But what I want you to do is make a high acceleration head motion, but not move very much. So just like a little like quick yes, no, yep. So similarly, your eyes stay on target. You could still see me while you do that or see the slide. And that's what this graph right here on the left is, the normal control. So your head looked like the blue trace where you moved really quickly to one direction. And then your eyes, you might notice I said equal and opposite, and these look equal, but they're inverted because this is for clinicians, so it makes it look easy. So you can see that it's a one-to-one mapping. They should perfectly overlap. And this is about as good as we get with goggle tracking. So that's a pretty good one-to-one head-to-eye motion. This is a normal subject. This is subject 9 when he came in before his operation. He's got about no eye movements for the 3 different planes of motion that we look at. That's really normal of someone with bilateral vestibular problems. Here's with 2 weeks of device use. So now we actually have something in each of the 3 canals. I see the bell. And here is with an acceleration term. So we actually, if I pan quickly back and forth between them, you can see we actually move the responses closer to the head motion. So here's all 3 time points shown on the same graph. The black dot is where they actually like start in time the head motion, and we're trying to just get that closer so that the eye and the head overlap. Thank you, everyone. Thank you, funders. Lots of pictures, and I'm done. Oh, I didn't need to stop the share.
Thank you. Thank you for that. Okay, wonderful. So we actually have quite a few questions in the chat. So we'll start off with Alan. Alan, would you like to ask your question yourself?
Oh yeah, sure. I was just wondering how your device encodes the different head motion speeds. Like, is it like pulse frequency, amplitude, etc.?
Sorry about that. I actually think I have lost my connection for a second. Could you please repeat that?
Yeah, I was wondering how the device encodes for different like speeds of head movement. Is it like pulse frequency, amplitude, etc.?
Both. Yes.
Both?
Yes.
Okay.
Yeah. So we have that. I'll go back. We have this mapping. There we go. Kind of blew through it, but this panel I actually has pulse rate up here and current amplitude here as a function of head velocity. And we actually do an activation process where we test people's thresholds and figure out what's the best mapping for them.
Okay, thank you.
I think we had another question from Thea.
Hi, Andrana. Great talk, as always. I was just curious, you were showing that users with a prosthetic implant for up to 6 weeks seem to be learning how to use their implant signal better and showing a slow improvement. So do you have any idea, maybe neurologically, what might be going on to cause that slow improvement?
Oh yeah, and there's actually even a slower improvement. Like we have one subject who maybe even at 6 months was kind of okay in terms of her efficacy measures for eye movements, but at 1 year said that now that I really spent my time, really learning how to use this, I can use it much better. And now she's riding her bike. So I think there's actually a lot to be said for the central nervous system circuits and basically practicing living with the device and like learning how to integrate your new vestibular sensation with an eye motion.
Really interesting. Thank you.
And Alex, I believe you had a question as well.
Yeah, I just wanted to ask, you talked about how there's a mapping procedure that happens quite early. How long does it generally take? I guess you sort of answered that in the last question, that it's variable. But
I'm really sorry. It's my AirPods that are cutting out. Could you just say the second half of that sentence, the question again? Oh,
yeah, I just wanted to know how long it takes to run the kind of mapping training procedure. You sort of answered that in the last one, but do you have a general?
Oh, gotcha. Well, there's 2 parts to that question. So there's how long does it take to acquire the data that we use and how long does it take to analyze the data that we use? And currently the process is it takes approximately 1 day or guess I got it down more to like 5 or 6 hours to acquire the data to get the actual thresholds, because we don't want to cause them undue pain, and pain might be a high threshold that we reach. As for analyzing the data, that's normally me and MATLAB until 3 in the morning. So I'd say approximately another 6 or 7 hours for a
That's
full
really
analysis.
fast. That's
I'm trying to get it faster. I'm trying to get it faster.
Awesome.
Move away from MATLAB. Yeah.
Not always the thing to do. Okay, so I think just because we are 2 talks behind and we don't want to go too over time, we'll go on to the next talk. But thank you, Adriana, and thank you everyone for all your amazing questions. So now we want to also let you guys know we're moving from 5-minute talks to 10-minute talks. So if talks seem a little bit longer, that's why. Our next presenter is Dr. Kartha, and she is going to be presenting from Dr. Dan Len-Lee's lab. And just as another mention, if you have questions, you don't have to type your question out in the chat. You can just put Q and we can call on you in the order you have your question, whenever you're ready.
All right, thank you. Can everybody hear me?
Yes.
We can.
Okay. So what I'm going to talk about is functional vision assessment in people with ultra low vision. And I'm a postdoctoral fellow in the ultra low vision lab working with Dr. Jalan Danieli. A little bit about, first, let me give you a little bit idea about what ultra low vision is. Ultra low vision, or ULV as we call it, is profound to severe visual impairment where the visual acuity is less than 20/1600. What that means is a person with normal vision, what they see from 1600 feet, a person with ULV will see it only up close at about 20 feet or so, or even sometimes they do not see that. So it also means that there is no form vision where they cannot see any details like reading letters or recognizing faces. But what they have is light perception. They can differentiate between light and dark, or even moving shadows or silhouettes. Currently, the most recent estimates from WHO shows that there are about 36 million people worldwide who are blind, which also includes people with ULV. That also means that these are the potential candidates for various treatments like stem cell therapies or gene therapy, or even visual prosthesis that Soo-Hyung mentioned a little while ago. So what we need when we have treatments is to measure vision before and after treatment to assess progress, right? So currently there is no standardized way to assess vision in people with ULV. And that's where my presentation comes in, and our work in the lab contributes to that. So just to show you a little bit about the vision testing and how it works, this is how we test vision normally. This is the standard of care, or the gold standard, where people are asked to read letters on a chart and up to a level where they can read. But for people with ULV, this is how it will look. It will look. So they cannot see any letters. It'll be all blocks. Like I mentioned, there is no detailed vision. But they do have some vision that they could potentially use for things like, locating a window in a room. Even though it looks like this, they can still say that. Or even like lights in the hallways, which would guide them to see the exits. So they could use their vision for that as well. Or even finding the crosswalk, even though they can't see the lines, they can still keep themselves within the lines so that they don't get into the traffic. So right now I'm going to demo a test that we developed in the lab. It's all in virtual reality because this is not safe or practical to do in real world. So we developed tests that can be done in virtual reality. I'm just going to stop sharing here and go into the test that we have. So can everybody see that?
We can see it.
Okay, so if you look in the small window, you will be able to see Roxana in the lab, and then she's going to walk in the lab while I'm doing this, so you can see the real world and the virtual world combined. So this is a street crossing scene, and her task is to actually cross the street safely by judging the stop gap when the car stops, and also to avoid the other pedestrians. So
just to let you know, we can't see the second screen of your lab.
Okay.
So if you unmute and use that as your audio, we'll see you. Yeah, perfect.
Great, we can see it.
Okay, all right. So I'm going to start it, and she's going to start crossing the street. You can start. Okay, that's the curb on the other side. All right, that's great. The next one I'm going to show is a scene where there is a cafeteria, and then it's a crowded scene with chairs and people walking around. What she needs to do is to meet the cashier, find the cashier, meet the cashier, and then come back and pop some balloons. So you will see that. Yes. There you go. Sorry. All right. Well done, Roxana. Thank you. And the next one I'm going to show you is a scene of a metro station. And then we are going to assess how she can safely get into a train, find an empty seat, and also at the same time avoiding the other travelers. That's the empty seat. That's great. Awesome. Thank you. All right. So basically what we are trying to do now is to work with people who have real ultra low vision and also simulate this in people with normal vision to learn how to calibrate this. We will be having different measures like response times and also the amount of veering that they do when they cross the street. And also we'll scan their head and the movements to judge how much they are off track and things like that. So we are currently doing this. Thank you.
Awesome, that was very cool. Ehsan said in the chat, this looks like Men in Black, just need some aliens to make it perfect. So with that, we'll open the floor to questions. Does anyone from the audience have a question? Not, do any of the judges have any feedback? Oh, Alex has a question.
I have a quick question. Do you find that patients with ULV have any consistency with their ability to see color? And is that an element that you could use in your platform?
So that's a very good question. So people with ULV can have different kind of conditions. Actually, it's not one particular disease. So depending on the cause, some of the retinal conditions, they do affect their color vision. So like you asked, it's not consistent. Different people can have different ability. So we cannot keep that as a, judging element, but some of them might be able to use it. We have had like some other modules like similar to this, where we have used some color contrast, so for some of them it helps, but it's not like, the main criteria. Yeah. Thank you.
There's another question from Celia.
Yeah, Celia.
Sorry, Gomes.
No, you're good.
Hi, yeah, my question was kind of related also. By the way, I really enjoyed the talk. I was wondering, is there a reason why, for instance, in the cafeteria simulation and the metro subway simulation, the contrasts were so dark, for instance, for the walls? I noticed it was black or really dark. And would this have an effect into how it actually assesses them being in those situations in real life where, for instance, when you walk into a metro station, the walls can be white, so very low contrast. Yeah.
That's great, actually. So yes, so one of the main problems that people with ULV have is working under low light conditions or when there are sudden changes in lighting levels. So we have, like I said, this is one of the modules, but we have different, things that we are assessing. So we had a first part where we actually tested people with different contrast levels to just get an idea about how their ability changes when the lighting changes. And with this particular scene, like what I showed today, what we were trying to do was to create different difficulty levels by introducing clutter. So, like in crowded environments and things, even though the contrast is the same, they do have issues navigating. So that was like particularly focusing on navigation. But we did have other parts to it, which I couldn't include in today's talk, but we did have other ones where we looked at contrast and eye-hand coordination as well. So we are testing different parts of this actually, yes.
Thank you.
And a real quick last question from Jean, and then we've got to get to the next talk.
Okay, so the question I have, so this is, I guess the point of this simulation is to be able to assess the person's ability to sort of perform in real life situation. But real life situation also includes many other senses like touch, like hearing, like smell. And so the question would be, do you intend to incorporate these somehow, and how would you?
So that's like a great question, and it's something that we are trying to see how vision changes influence their performance right now. So particularly these tasks were developed without any other feedback like tactile or auditory feedback, only to see how vision affects, because right now we don't know about that. But at the next level, yes, definitely introducing the other senses, a multi-sensory way. If we have to rehabilitate them, I think that is the way to go by, because by introducing the other ones, we can see if it improves performance. But right now, we specifically decided to exclude the other ones only to see how we can actually, assess their visual performance. Yeah, so that's another advantage of virtual reality. We could add in or take off, noises or other, modes basically. So yes, thank you.
Great, thank you, Dr. Kartha. It's a really great talk and very cool demo. So our next talk will be by Dr. Salimpour from Dr. William Anderson's lab.
I guess I can share my screen now. Can you see my screen now?
I think it's starting.
Perfect.
Now we can see it.
Now we can see it.
You can see now, yes?
Yes.
Okay. Okay. So thank you so much for the opportunity. My name is Yusuf Salimpour, and I am assistant professor at neurosurgery department. First of all, thank you so much for having me today, and I am very glad that I'm not going to compete with great students and with such a talented student and great presentation. So today I'm going to first introduce ourselves and also the study that we are working on for now. So we are focusing on the idea of phase-dependent neuromodulation system. We believe that this kind of stimulation has a lot of potential to be next generation of the brain stimulator to treat neurological disorder. So most of you are familiar with the neural oscillation, and you know that mostly it's limited to the frequency band that we are interested. Like in the beta band, mostly focused on the motor network and also the sensory motor network and the motor planning and execution. But we know that even this limited observation to the brain oscillation can show in the brain disease as well. So in the case of Parkinson's disease, this beta band is exaggerated, and human an actual Parkinson's disease, and also in the animal model of the Parkinson, we can see how in the beta band power, the beta oscillation is more than a normal behavior animal or human. But it sounds that the oscillation is not very simple. It's not that simple that we are seeing. It could be more complicated than that. So on the right hand side, you can see that in the well-controlled experimental data, how the low frequency oscillation could couple with the high frequency. And you can see that in the specific phase of low frequency, the high frequency start to emerge. In the left side, you can see one of examples from our intraoperative test. And here you can see even more detail that how in the human motor cortex, in the case of the Parkinson, the high frequency, which is a gamma activity, is tended to be more exaggerated in the peak of low frequency oscillation, which is the beta frequency here now. It sounds that whatever we are observing is not just a recording, but it reflects a more complicated phenomena in the brain. We don't know what is going on that we record such a an coupling into frequency, high and low oscillation. In the top, you can see that this is a high frequency gamma band recorded from the motor cortex. And you can see how it's coupled to the beta band oscillation. And in the lower panel, you can see how this gamma can be reflected single unit activity. So it indicates that the phenomena is very complicated, and what we observe as a decoupling could be either projection of the gamma activity or single unit activity, or maybe both at the same time. So how we measure this complex phenomena, it's not that easy to quantify, but we use a modulation index, which is something acceptable. We are not saying that is a perfect, but it's acceptable the way to quantify. I don't want to go through the detail because of a limited time, but we can see that in brief, we decompose signal to the low frequency, phase, and high frequency amplitude, and we see that in which portion of the phase these two are coupled. This is a simple example. On the right-hand side, you can see how the left-hand side modeling like a 10 hertz oscillation with the 150 oscillation coupled in the peak can be reflected to the modulation index. And in the modulation index, you can see that there is a coupling in 10 hertz between 150 hertz. So this is the way we can represent this phenomena. The coupling, although it reflects some healthy normal activity of the brain, but it's correlated to a lot of neurological disorder. And this list keep increasing for now based on the recent data. But for now, we are focusing, I'm going to show you some example from the Parkinson, but it's not limited to Parkinson, and it covers more neurological disorder as well. Like in the Parkinson, see, when the patient with the Parkinson, you record from the motor cortex, there's exaggerated coupling. This is a modulation index that I introduced in the previous slide, but you are not seeing the same thing on the dystonia or on the epilepsy. And it shows that it's kind of like a biomarker for the movement disorder. So we know that we have two acceptable clinical treatment for the Parkinson. The famous one is the medication, and you can see if you put the patient in the medication, the exaggerated abnormal coupling goes away, and if the patient took the medication aside, this baseline activity back to the position. So it says that effective treatment need to reduce this activity, and at the same time, the next acceptable clinical intervention for the Parkinson is DBS. And in the case of DBS, you can see that recording from the motor cortex shows exaggerated coupling in the baseline. And if you turn the DBS on, you can see this coupling goes away. And if you make it off, it backs again. So any effective treatment need to reduce this coupling. Our idea is that why not we intervene directly, and why not modulate this an abnormality of the brain directly, and then see how it's reflect to the symptom of the disease. So our idea is that let's fix the network directly rather than indirectly using medication or the brain stimulation. So the idea looks simple. We try to lock to the either peak or trough of the low frequency oscillation and try to modulate the amplitude. Although it looks simple, but it has a lot of technological and technical challenges. Like if you look at the definition of the phase, you can see that this is not the easy quantity to measure, and you cannot monitor the real time because you need to know the future if you want to know the phase now. How we can resolve this issue? Imagine that you are at time 0 now, and you want to know that what is the phase now. You need to know the future, and you do not have it. We do the prediction. So we predict the future combined with the measure up to now and try to estimate as accurate as possible the phase for now and estimate the next desired phase could be either peak or trough. So this is an example how we predict the signal. It could be like an either one dimension or more complicated 2-dimension way of like an estimation. This is a summary of what we are doing in the operating room to detect the phase and to find the target and the stimulation. I don't want to go to detail, but you can ask me offline or later on any question you have about the detail of this technique. But so in the operating room, we take the advantages of the DBS and slide a temporary grid electrode In the operating room, we are blind. We don't know where is the motor cortex, but we use the idea of the phase amplitude coupling to detect the motor cortex. Later on the left side, you can see this is the anatomical offline process, and right-hand side, this is online intraoperative process. So it sounds that the detection of the cortical structure, especially the motor cortex, is kind of in agree with the anatomical information. So our electrophysiology is in agree with the anatomical measure, which comes from the neuroimaging. This is an example how we see exaggerated coupling in the motor cortex and not in the other areas that we are recording in the operating room. So we find the target. Now we set our stimulation to either peak or trough, then we run our algorithm. So this is one example. Like in the operating room, patient is at rest. We start with the baseline recording, then turn on the machine, do the peak stimulation, then another an after peak stimulation, then trough stimulation, and then again after peak. Although we shuffle this for the population, but this is a general idea how we run the experiment. So here is the two example. In the top, you can see that we lock to the phase in the trough. But if you want to know that how is our accuracy, it's not very difficult. Just wait a few milliseconds, you can see the actual phase. So if we predict it now, in the few milliseconds later in the future, we will see the actual phase. So we can always evaluate our performance. So the top one shows that we stimulate at a trough. The bottom one shows we stimulate at a peak. And it sounds that we are kind of accurate because the average, the mean is around the target phase that we set the device. This is an example how in the operating room, we turn on the machine, detect the peak, and start the stimulation based on the peak. The lower panel is not the time frequency, is the dynamic pack modulation. It shows that how dynamically the phase amplitude coupling monitored across the time. So as you can see, when we turn on the stimulator on, after a delay, the exaggerated packets start to show, which shows that this is kind of an effective. This is a quick example how it works. And based on the peak and trough, we can either modulate or reduce. And this is the average of our 5 subjects so far, how we can increase or decrease the pack. And I guess I'm running out of time. So with that, I would like to thank our collaborator in the neurology, in the epilepsy, and in the neurosurgery that we are working on. Yeah, I guess I'm ready for any question.
That was great. Thank you, Yusuf.
So much.
So could we get a question from one of our judges first before we jump into some audience questions?
Yeah, on the when you looked at the PAC and meds, you had complete disappearance, complete removal of any of the signal. But on the DBS, you said it disappeared, but it didn't. Do you know how, was that still effective in that situation, or?
Actually, that's a great question. That's the reason when you put the patient in DBS, you need to have a medication in line as well. So DBS by itself is not a treatment. DBS plus medication is effective treatment. But you are absolutely right. It says that medication is more effective than DBS. But consider that some patients are medication resistant. So in that case, definitely you need another solution that we are working on.
Okay, thanks. Great,
thank you. And then we have a question from Alan.
Hello, I really like the talk. So one thing I was a little confused about is the stimulation trying to interrupt that high frequency activity, or how does the stimulation stop Parkinson's symptoms?
That's a great question, and I put a lot of detail aside. That's the reason that you are maybe a little bit confused. But the reason, actually, we are going to network in the brain is not normal now, and we are going to normalize it. It not necessarily reduce the high frequency activity or not necessarily increase it. It just decoupled the high frequency with the phase of low frequency, which is exaggerated and abnormal.
Okay. Thank you. That makes a lot of sense.
That's a great question. Last question from me. So I know your patients are in the operating theater. I know that they're anesthetized. I know they're probably also on their dopamine medication in the theater. Do you get a sense, and you said that they're resting, do you get a sense if patient movement modulates the phase amplitude coupling. So I guess your patients are resting, but if you were to have them do simple repetitive movements, do you think
Again, that's a great question. Actually, patients are a little bit again mildly sedation. They're awake in the operating room, and so I did not include all of the data we have so far. We monitor the symptom intraoperatively, and what you're seeing here, like the result of like an exaggerated coupling, is a very wide time window. Movement is a very short time window. I guess that like an fluctuation is inside that wide time window. So it's kind of like goes away. The effect goes away if you widen your time window. But we have like some data shows how the hand dexterity improve if you do this simulation intraoperatively.
That's great. And actually, that's perfect. That was 3 minutes of questions, so we can jump to the next speaker. Thank you, Yusuf.
Stop my sharing. Thank you so much.
Awesome. So we do have 3 more speakers left. So with that being said, we may go over our time. So if you have to leave early, we completely understand, but hopefully, like, I'm enthralled. All the talks are amazing, so you don't really mind if we're going over. So hopefully you guys feel the same way. Our next talk will be by Rulan Sun from Dr. Adam Charles's lab.
Let me share my screen. So, oh, sorry, wait a minute.
No worries.
So can you see it now?
Yes.
Okay. Hi everyone, nice to meet you all. I am Roland Sun, a first-year BME PhD advised by Dr. Adam Charles. The theme of my today's presentation is decoding functional ultrasound signals via sparse representations. There are many methods to image brain. Some methods aim to image neuronal activities directly, like calcium imaging and the physiological methods. There are also indirect methods. In brain, the neurovascular coupling in the vessels surrounding the activated neurons will induce hemodynamic changes. More oxygen and more blood are needed when neurons are activated. So the indirect methods aim to image such hemodynamic changes. Functional ultrasound is such an indirect method. It's based on Doppler effect and detects the variation of cerebral blood volume over time. Recently, Dr. Carl Anthony's group in University College London also published a new paper and gave detailed proofs that functional ultrasound signals can reflect neural activities accurately in time and in space. There are multiple advantages of functional ultrasound method. Firstly, with the assistance of ultrafast imaging technique, functional ultrasound achieves high spatial temporal resolution. Depending on the ultrasound frequency used, its neuroimaging has a typical 50 to 200 millimeter spatial resolution and features a temporal resolution in the tens of milliseconds. Secondly, functional ultrasound enables whole brain imaging. It is unbiased to certain area or certain cortex, can record large areas of the brain, including deep structures. As we know, complex behaviors need coordinated activities of many brain regions. So the whole brain imaging can help to discover new areas previously unknown to be involved in many classic behaviors. Additionally, with just an ultrasound probe, functional ultrasound has high portability, which is different from MRI that requires objects to stay still in a big machine. So we can record signal from freely moving rats. The expanded spatial temporal scale and the ability to perform imaging in freely moving rats enable functional ultrasound to capture dynamics in the whole brain in more naturalistic behaviors like decision-making. The decision-making task in our work was developed by Dr. Ahmed Hadi in Princeton University. The task is named as ultra-long evidence accumulation. At each trial, an LED light indicates the trial start. The rat then uses the center nose poke to initiate the trial, wherein an auditory Poisson click train is played on left and right sides.
After that, the rat uses the left or right nose poke to indicate a selection about which side has more clicks. Water rewards will be given for correct choice. Poison clicks here in this task play for 3 to 5 seconds. The length of the trial duration matches the timescale of hemodynamic changes and allows functional ultrasound, the large-scale imaging technique, to capture activities across areas. With functional ultrasound data, what we want to decode here is which frames are in the clicks given period and which frames are outside the C in, C out period. In functional ultrasound raw data, there are more than 10,000 pixels in one frame, which will slow down the decoding algorithm. So to decode the signal more efficiently, we reduced the dimension of the data with sparse representations. The algorithm here was originally developed for calcium data, but we applied it here. The main idea of sparse representation is to factor the data cube into a set of spatial profiles and the corresponding set of time traces. The spatial profile means the location within the field of view that a component occupies. The component can have different definitions in different imaging modalities. For example, in calcium imaging, a component represents a neuron, but in functional ultrasound imaging, they are not neurons. So we can assume that some co-activated regions in the brain is a component, and the time trace is the averaged signal for a component. For neuroscientists, the main interest lies in time traces, since they are the variables linked to behavior and stimuli. So in our algorithm, time traces are the dictionary, while spatial profiles are sparse coefficients. Thus, we model temporal traces directly, places more importance in finding temporal activities, and treats the spatial statistics as secondary. Here are part of our decomposition results of dictionary that has 30 components. We visualized the spatial profiles of these components in Brain Atlas, and we can see that these components are scattered in the whole brain. Decoding the functional ultrasound is just a classification task. When we judge whether a frame is in the clicks given period, we also consider the past n-1 frames, since the variation of the signal over time can offer more information. The formulas are shown here. If we take the raw data as the input, the dimension of the decoder will be nm, where n is the number of frames and m is the number of pixels in one frame. But if we take the decomposition results as our input, the dimension of the equivalent decoder will become nr, where r is the number of components in our decomposition result. Since the number of components is much less than the number of pixels in one frame, the complexity of our equivalent decoder can be much smaller than the original one. We used SVM to train the decoder. We firstly used the linear kernel, but the results were not good. The precision is always lower than 0.7. Then we found Gaussian kernel. The nonlinear kernel has a much better performance. All indicators have higher scores, and this may mean that the relationship between brain activities and the whole brain cognitive variables is nonlinear. We also compared the precision of results when we used different number of last frames and a different number of decomposed components when decoding. We can see that with the increase of the number of last frames, the precision difference among different components will decrease. In order to test the robustness of our decoding algorithm, we also decode other parameters. For example, we also decode whether a frame is during the period when the rat made a decision and hit the side poke. And we also decode whether a frame is during the period when the rat got water rewards. With the nonlinear decoder, we can see that the results are still good and the precision is always around 0.8. Above all, we have processed the functional ultrasound signal via sparse representations and reduced its complexity. And we also decoded the decomposed signals with a nonlinear classifier. In the future, we will try to give more explanation about the components in the decomposition results, like the reason for some regions are classified as one component, and if some components are overlaid with some known important regions in brain atlas, and with some computational network models, we will also try to model temporal dynamics of functional ultrasound components over time. We can see that functional ultrasound is an emerging imaging method in neuroscience. So our successful try to decode the functional ultrasound signal can help to its further application in neuroscience community. That's all about my today's presentation, and thanks for your listening. Thank you.
Thank you very much. So let's get a question from one of our judges first. How about Dr. Vogelstein this time?
Nice, that was very nice. I wonder, you probably know about Eva's secret random projection Fourier transform thingy because you're both working with Adam. I wonder, would that kind of approach be helpful here?
Sorry, I didn't get your idea. Could you apply the method to what problem?
So you have this really high dimensional, spatially time varying signal, and Eva developed a method to do nonlinear dimensionality reduction. I just wonder if you can apply it here and if it would be helpful.
I think maybe we should have like how to define the component in different imaging modalities. Since her data is, I think it's calcium imaging data, I think maybe it can be explained easily with this method, because with this method for calcium imaging, there are neurons as components, and so that when we decompose the data, the spatial profile and their time traces can be explained more easily. And I think in functional ultrasound data, when we apply the algorithm here, it will be more difficult because there are no components in our data. So I think we can try to apply this algorithm to her data and take a try.
Thanks.
I think we have another question from Ghislaine.
Yeah, you mentioned that the spatial resolution is 5 to 20 millimeters. I was wondering if that should be 5 to 20 micrometers, because 5 to 20 millimeters would be the size of the rat brain, pretty much.
I just saw this data from a review for the functional ultrasound technique, but for more detail, how to achieve this spatial resolution depends on the ultrasound frequency you used. And in functional ultrasound imaging method, it uses plane waves to scan the area. So I think it highly depends on the ultrasound signals you used. And for more details about many technologies, since I'm not very familiar with that, I just read it from a review paper.
Okay, thanks.
Actually, that leaves us just about ready to go on to the next talk. Thank you, that was great.
Thanks.
Wonderful. Our next presenter is Roxana Sadeghi from Dr. Dan Legley's lab. Roxana, whenever you're ready.
Hello, my name is Roxana Sadeghi, and I'm going to tell you about, I'm a fifth year PhD, BME PhD student working with Dr. Jolan Danley, and I'm going to tell you about visual perception with visual implants. But before that, I'm going to review the visual system. So the lights goes, the light hits the lens in the eye and goes to the retina. And then in the retina, we have we have light sensors, which we will call them photoreceptors. And when the light hits the photoreceptors, they will activate the neurons inside the. Let me make my cursor as a laser. I forgot how to do that. Oops, no. Sorry. Yeah, okay. Can you see my pointer here?
Yes.
Okay, so then once the light hits the photoreceptors, it will activate the neurons, and the neurons send the signal to the brain through the optic nerve. So the signal goes to the optic nerve to the thalamus, an area called LGN, and from LGN to primary visual cortex, and from primary visual cortex, it goes to the higher areas, and it will create the visual perception for us. If in between of this, in the visual pathway, any of the neurons get affected by the disease and become dysfunction, then the signal won't go to, the signal cannot pass through to the visual cortex and to the higher areas, and it will disrupt the perception. For example, imagine that we don't have the photoreceptors and the lights cannot pass, cannot be detected by the photoreceptors. But there are still some neurons in there, but they cannot get any input from the photoreceptors and nothing goes to the brain. So in many forms of blindness results in the neural damage, which is not reversible with the knowledge that we have. And there's no cure and treatment for that. But the visual prosthesis can be an option for the treatment. We know that electrically stimulating neurons in the visual pathway will create visual perception. We can put electrodes, for example, in the example that I told you in the retina that the photoreceptors are died, we can put electrodes there, and the electrodes can be act like the photoreceptors and stimulates the neurons that survived from the disease in the retina and because the thalamus and the cortex can stay normal, it will, so the neuron signal can be sent to the brain and we can create the perception. So in the visual pathway, we can stimulate any neurons in the visual pathway, retina, optic nerve. We can have a deep stimulation of the thalamus and the visual cortex, of course. How the general concept for the visual prosthesis is the same for all of them. The patient or the user will wear a pair of glasses that has a camera in the middle. The camera captures the image, and then the image goes to the video processor unit or little computer that they're going to carry on. And the video processor unit will assign each electrode the correct pulse parameters and send those information to the implant via a wireless connection to the implant. The only visual prosthesis with the US FDA approval for like at the current stage is Argus 2, which is the periretinal implant. Second Sight Medical Products company built this implant, and there are more than 300 blind individuals using this implant worldwide. It has a 60 electrodes in a 6x10 electrode array inside the eye. And just a fun fact, normal human eye has 126 million photoreceptors. And the electrodes are pretty large surface electrodes that sits on the tissue. And it will requires functioning neurons in the eye so it can send the signal to the brain. And it can cover about 22 degrees of the visual field. Just to give a sense of how the 22 degree is, if you stretch your hands in front of yourself, that would be about 22 degrees. So what an Argus 2 users can see, the yellow box is the visual field, about the visual field that they're looking at. And in the video, the person will move to the left and right, and you will see the perceptions on the right side. So they can see only because of the disease, and large electrodes, the stimulation will spread out and they see very blurry image. They can see moving shadows and they can point to the lights, but that's about it. For complete blind subjects, this is also very useful vision. It can be very helpful for the mobility and orientation, but still it has some limitations. So imagine that the camera sees a view like this, and there is a clutter background. It's not that simple background that as before, and there are some lights in the ceiling, the door is white, the background, there are some other objects in the background. And they want to, the subject, the task is to find this person, but what they see is something like this. Again, it's very blurry. They cannot tell the shape or any other details. They don't see colors, and they cannot distinguish objects. They cannot tell if it is close by or further away. But imagine that instead of using this camera input, we only shows the target objects that they want to look for it, and just remove the background. Then the perception will be much easier to understand. So for example, here we show only the person, and what they perceive is a light only from the person. So they can detect the person easier. How we can do this? We can use the thermal camera, which only shows the warmer objects in the room, for example, the person. If they want to ask for assistance in a public place, or they're, for example, they're preparing a meal, or anything like a hot cup of coffee, they can use the thermal camera. And also we can use a distance filter. So the distance filter has two cameras on the side of the glasses, and based on the camera view of these two, we can identify the object's distances, and we can only shows the objects within a certain distances. So we can only remove the background and show only the objects that are close by, which will be very helpful for orientation and object avoidance. So we test our system in a different task, but I'm going to show you the example of the mobility performance with the thermal camera here. The task is, we were on the hallway, the subject was in the middle of the hallway, and the task was to find a person standing at 3 or 5 meters. And let's see how he's doing. So I'm standing about 5 meters away from him. So he started looking, he saw the doorway, and he thought that as the person, because that was bright. He reached out his hands and scanned the room and finally found the target at the end. But here's a performance with a thermal camera. So he kept looking, he saw the target, and he directly, boom, went to the target. So the performance is better with a thermal camera, just finding the person in the room. And this is the results with the 4 other Argus 2 users. And it shows that like they could complete it with a normal camera, no filter camera, they completed 56% of the time, but with a thermal camera, almost all of the trials was completed successfully. This is another example for the object avoidance using the distance filter camera. So the task is to walk to the end of the hallway without hitting any objects, and there are 3 objects either hanging from the ceiling or on the floor. So this is the example with a normal camera. He didn't see the first one, he hit the first one. He thinks the second one is a doorway, so he hit the second one too, but he didn't notice the third one, but he didn't hit it. And this is the performance with a distance filter camera. Again, there are 3 objects in the hallway. So he saw the first one, goes around the second one, and he sees the third one. And if you notice the performance with the distance filter, he's more like chilled. The hands are in the pocket, and he's like, I know where the objects are, and I'm confident to go. So I like that. And the results also with in total 3 Argus 2 users shows that with the no filter one, they detected 12% of the time, and they hit the objects 48% of the time, but with the distance filter, they detected about 95% of the time, and they hit.
10 minutes.
Okay, sure.
Oh, you were close.
Yeah, so the summary is the electrical simulation creates the visual pathways. The current technology with the visual prosthesis has some limitations, but still within this limitation, we can improve the performance using other like cameras, like a thermal camera or distance filter. Yeah, thanks.
That was excellent. Thank you. So let's start real quickly with a question from one of our judges. Maybe we could do Dr. Montague this time.
Yeah, with the Argus, is it possible to integrate a distance filter and sort of some sort of imaging directly into that? So that would be able to be incorporated in the current technology.
Yes, yeah. So what we did was similar. So let me show you the results. Like, cannot go back. Okay. Well, all right. Never mind. So the glasses that they're wearing, we can have two cameras on the side and just send that input to the VPU instead of the normal glasses. So yes, we can include it in the current version. But right now we just tested in a lab, and like the next step is to just give the device to them and take it home. They will take it home and test it at home at different like situations that they might need. And the idea is that then they have the ability to switch between the modes. So it's not just only the distance filter only use the thermal, but switching the modes can be helpful, for example, to distinguish the person from the chair, or with the thermal and the normal filter, or we can use a distance filter when they're walking and then they switch to other modes when they do.
Well, it's a possibility to introduce different modality. I mean, you could flash something, so you could have both modalities at the same time. You wouldn't switch.
Right. So we can incorporate the thermal camera with a distance filter. So we only showed, for example, the hot objects within certain distances. That also is possible. But for different needs, they might need different modes. So it will be good to have this switch mode as well, like the
I was thinking of the Argus, where they walked around and run it. If the images that were close were flashing, it would call their attention to it, for instance.
Exactly. Yep.
So they could use that directly, and you'd have both modes at the same time.
Right. Right.
Okay, thanks.
Yep.
Let's see, we've got a little less than a minute. How about a quick question from Geely? She's still here.
Hello, can you hear me?
Yep.
Yeah, go ahead. We can hear you.
Oh, yeah, I'm just wondering, how is the data from the video processing unit being transferred to the electrodes? Is it wireless?
Yes. So with a transmitter coil, it's wirelessly.
Okay, thank you. That's my question.
Great. So, okay, that was a lot of questions. Sadly, we don't have enough time, but maybe we can revisit at the end. So let's jump to the next speaker, the last speaker.
Yes, our last but definitely not least speaker will be Dr. Senarnatha. I apologize if that was incorrect, who is part of Dr. Pathak and Dr. Thakur's lab.
Good afternoon, everyone. My name is Tanaka, and I'm a postdoctoral fellow in Dr. Pathak's and Dr. Thakur's laboratories. So today, I'm really excited to talk to you about some of the recent work that we've been doing in developing this very small microscope that can image the brain's neuronal activity as well as its vascular functionality via optical imaging techniques in awake and behaving animals. So let me get this. Okay. So as most of you know, optical imaging systems are traditionally really large and bulky. And this means that animals usually have to be either immobilized or anesthetized to be imaged. And this actually alters the brain activity that we are supposed to measure. In addition, this immobility prevents us from correlating animal behavior with the measured brain activity. So the wide range of experiments that could be possible gets really restricted to only anesthetized or immobilized experiments. So in theory, most of these problems can be solved by miniaturizing or downsizing these large optical imaging systems, for example, to the size of a fingertip. Now, shown here on the left is an image of a typical benchtop imaging system. And on the right is one of our miniature microscopes drawn to scale. Now, this level of miniaturization solves almost all the problems that I just mentioned. However, these tiny devices have their own limitations. For example, most of the miniature microscopes that are available today offer only a single imaging channel, which tends to be a fluorescence channel. And therefore, these are usually used to look at neuronal activity by imaging GCAM fluorescence. However, these microscopes do not offer a vascular imaging channel. Conversely, these miniature microscopes that I've shown on the bottom offer several vascular imaging channels, but they are incapable of performing any direct neuronal imaging. Therefore, while this current crop of miniaturized microscopes have allowed neuroscientists to image brain activity in freely behaving animals, their inability to look at multiple biological variables is a severe limitation. So to address this, we developed a unique microscope with 3 imaging channels: fluorescence, intrinsic optical signals, and laser speckle contrast, which enabled us to look at neuronal activity or fluorescent labeled cells, microvascular structure, as well as blood flow in the brain. Now, here's a brief overview of our microscope miniaturized architecture. The bottom view here shows the arrangement of illumination sources for all the imaging channels, with the side view showing the rest of the miniaturized components. These allow optical access to the brain via a cranial window and a head mount, and can be controlled via a simple graphical user interface. The microscope is shown here next to a US quota and achieves a 5 micron spatial resolution while covering a 3 by 3 millimeter field of view, which in mice is sufficient to cover an entire functional cortical region. Finally, with strain relief, the microscope only weighs about 3 grams, which allows imaging in freely behaving animals conveniently. So first up, we validated the performance of our microscope by comparing it with a benchtop-based system. Shown here on the left are images of microvascular structure from the same field of view by each of the imaging channels of our miniature microscope. And shown here on the right are their counterparts obtained from a non-miniaturized standard benchtop imaging system. These graphs on the far right compare their intensity ranges, and as you can see, they have a very reasonable correlation. Now, with the performance of our microscope being validated, we then went on to use our microscope to look at neurovascular coupling in the auditory cortex of an awake mouse. So shown here in the bottom are videos of 10-second long epochs, averaged over 30 trials in response to a 4 kilohertz sound stimulus given for a brief time period. As you will be able to see on the video to the far left, which was obtained by using the fluorescence channel, imaging GCAM-bound calcium fluorescence activity, the neuronal response showed a spatially localized activity pattern. However, as shown in the video in the middle, which was obtained with the iOS channel, the cerebral blood volume shows a sustained vasodilation pattern that had a tremendously larger spatial footprint. Now, this mismatch between neuronal function and vascular function was amplified even more with the blood flow response seen here on the far right, obtained with the laser speckle channel. So this ability to investigate neurovascular coupling in its disparate temporal and spatial formats, which was not available in the current crop of miniature microscopes, we believe is a significant value addition to the neuroscientific community. Now, apart from neurovascular imaging, we can use our microscope also to study brain diseases in great detail. Shown here is an example where we've used it to characterize the alterations that occurred in a brain tumor microenvironment throughout the lifetime of the tumor. So the first row here was obtained using our fluorescence channel. And instead of looking at neuronal activity, here we are observing the progression of a 9L brain tumor, where the tumor cells had a GFP, green fluorescence protein marker. And the second row here shows the concomitant sprouting of new microvessels to support the heightened metabolic demand by the tumor. And finally, the third row shows the induction of new blood flow into this vascular territory, which you would notice lags by a couple of days the formation of new blood vessels to support the tumor growth. Of course, these trends can be quantified as well. And the final insert here shows the a zoomed in view of our images to show the final level of details visible with the microscope. So just like with neurovascular coupling, being able to observe the alterations of multiple neurophysiological variables throughout the life cycle of a disease model imparts tremendous value to understand and gain an overall picture of how the disease progression is happening. With that, I would like to wrap up my short presentation and thank my mentors, team members, and funding sources. However, our journey is just beginning, and we are looking forward for collaborators to test our microscope, fund building new prototypes and commercialization ventures. So if you are interested, do contact either me or Dr. Pathak on the below email addresses. Thank you.
Excellent talk. Beautiful images. That's really very impressive. So let's get a quick question from one of our judges. Maybe we'll go with Dr. Vogelstein this time.
Yeah, I have a question. So that was, that's really cool. The images are really pretty, and I understand the general motivation. I wonder, do you have an example of a very specific neurobiology question that your system is particularly well suited to answer, or in the other ones you don't think you would get as clear an answer about, just one specific question?
Sure. So one of the questions that we are trying to answer now is about seizures. So seizures not just have a neurological impact, but there is a tremendous vascular impact occurring together with seizures, and a lot of imaging systems only image for a couple of hours. So they don't capture the entire recovery after a seizure that happens in neuronal and vascular activity. So one of the projects that we are working on specifically is to image for 12, 24 hours continuously multiple neurobiological variables post-seizure to understand how exactly the recovery from seizure happens. And in such an instance, having a multi-contrast microscope or multi-channel microscope like ours and being miniaturized so that you can do awake behaving imaging is critical.
Thank you.
Let's see, we have a lot of questions from the audience. Maybe we'll go with Ehsan first.
Yeah, sorry if this question is naive, but what is the bottleneck for the frame per second, basically the time resolution? Can you increase it to higher? For example, for calcium imaging, it's usually typically 30 frames per second. Why your system is 15 frame per second, and can you increase it?
Yeah, so in theory, yes, but the current values were set because of several practical limitations. So I'll answer, there's two questions to it. One is spatial resolution and one is temporal resolution, right? So in terms of spatial resolution, it's always a compromise between what resolution you want versus what sort of a field of view you want to cover. So we can easily increase the resolution, but it would reduce the field of view that we can image. So that is very flexible with our microscopes. Here we wanted to cover a considerable large field of view, and that's why we set the resolution at 5 microns. Temporal resolution is slightly tricky because one of the requirements for a miniature system is to have like a miniature image sensor, and that places a lot of restrictions on how fast you can go about it. Now, there are several microscopes out there specifically focusing on calcium imaging, and they can go up to even 60 frames per second, the miniscopes. We are working on one of the projects that we are working on uses a similar image sensor, and we'll be able to get to that frame rate. But the types of image sensors that we've been using are incapable of going, let's say, beyond roughly 20 frames per second, given the number of pixels that we need to catch. So just to put an icing on the answer, if you can compromise on spatial resolution, let's say for calcium imaging, if you are interested in a 50 micron resolution and you want to cover the same field of view, we can easily reduce the size of each image we capture, and we can proportionately increase the frame rate. So we've tested, let's say, the images that I showed you are 640 by 640 pixels. We've tested a version with, let's say, 128 by 128 pixels, and we've been able to get about 40 to 50 frames per second. So in a sense, it's an advantage that we have. We are able to sort of modulate the spatial and temporal resolutions as we want to suit our application. And I just presented you a certain fixed set of them.
Great. I think we'll pass it over to Cynthia now.
Hi, everyone. Janica, could you stop sharing your screen?
Yep.
Cool, great. So today has, I think, gone even better than really anyone on the TNT board could have even imagined. These were just amazing talks and demos, and it was so exciting. So first, while I have your attention, we get to the audience vote. I just want to remind you guys that we'll be voting on the best winner, the best audience favorite for today. But you should come to day 2, which is going to be April 13th from 3 to 4 p.m. EST, maybe with some overspill, to choose the overall audience favorite between who you choose today and the next day, and to hear some more really exciting talks. We also had so many great questions. So the TNT board has already agreed that Celia Fernandez is one of our best audience winners. But based on all the great questions, I think we're going to be giving out even more prizes. So we'll contact you soon. And then besides our day 2 schedule of events, the other event that's coming up, if you guys are really interested in neuroengineering, is that we're also going to try to be putting together a neuroethics panel. So we invited 3 ethicists from across the US who specialize in considering ethical implications of different different neural implant designs and how basically patient experience and design play into these different questions. And so if you're also interested in those things, please also look for our event May 20th and feel free to attend that. So besides that, finally, if you want to keep following what TNT Network is up to, please join our listserv or follow us on any of our social media. So after that shameless plug, we are now going to turn to the polling. And so it's your chance to tell us who you thought had the best talks of today. So if everyone can see the polls, please go ahead and start voting. Oh, I don't know if you guys can see what I can, but I'm seeing the numbers come in. So far, we've got about 18, 19 votes. We're getting up there. Oh, it's close. Wow. Okay, about 75% of you voted. Or we can get any more votes. I guess maybe some of the attendees can't vote, so we might be reaching a cap soon. 83. We have 37 out of 42 votes, which might be everybody not considering the judges.
Is that enough votes to swing the results?
Let's see. We do have a lead winner actually, so that I think is, yeah, that is enough. Okay, there we go. So we have a pull ahead winner. So congratulations, Andriana. You pulled ahead with some close competition also with Janica. And so you'll be in the running for day 2 to win the audience favorite. So thank you again, everyone, for coming. This is really exciting. And also presenters, please send us your presentations if you can, so that the judges can also look at them when trying to do all the scoring. Thank you, everyone, for coming today, and hopefully we'll see you on day 2. Bye.
And to all our speakers, thank you. You all did excellent, excellent job. Thanks for making that a success.
Hello, everybody. I'm Cynthia Steinhardt, and I'm the president of the TNT Network. And I have the great pleasure of welcoming you to day 2 of the TNT Showcase. So for anybody who was here on day 1, welcome back. And for people who are joining us for the first time, if you're curious about what you missed, we have the video up on the website right now for you to see. So before we got started, if anyone's curious what you missed, we had 9 amazing presentations on day 1, and we voted for the day 1 audience favorite at the end. So today, again, we're going to be joined by our 3 judges, Jean Friedman, Associate Professor of Otolaryngology and BME, Dr. Chuck Montague, BME Industrial Liaison, and Joshua Vogelstein, Professor of BME of the BME Department. Thanks again, not only to our judges who are going to be sharing their expertise in translation and presentations, but also to, of course, our MCs. So day 1, our MCs were Gulnish Kamali and Alex Weiss, 2 of our TNT board members. On day 2, it's going to be me, Cynthia Steinhardt, and Esad Najad, who are 2 other board members who will be our MCs. So for anyone who wasn't here the first time, the way it's going to work is we're going to have 5 more amazing talks today. Each of them are going to be 10 minutes. And the goal here is really to prioritize connections with the audience. So how it's going to work is exactly at 10 minutes, we're going to cut you off. And there'll be 2 to 3 minutes where we first take audience questions and then turn to our judges for feedback. And then please stay at the end, because you're not only involved in participating and talking with everybody, but you also have a vote in who can win the day 2 audience favorite prizes. And finally, because it's our tradition and it's the first event, we're probably going to go a little bit over time. So how it's going to work is we're going to bleed over, try to stay to the end. And then because the competition was so close on day 1, what's probably going to happen is we're going to hear back from the judges, maybe by Friday, about who all of our winners are going to be. So with that, I think we can get started.
Our first speaker of the day, I'm honored to introduce Grace Foxworthy from Dr. Jean Friedman's lab. Grace, please go ahead, share your screen.
Can you see it now, the presentation?
It's a little small.
Yeah, there.
Perfect, go ahead.
So hi, everybody. My name is Grace, and I am a PhD student in the Biomedical Engineering Department, and I'm a member of the Machine Biointerface Lab. And so our lab is concerned with pretty much everything to do with direct current stimulation. So we have some people working in lab on exploring potential applications for direct current stimulation via modeling or in vivo electrophysiology. And then kind of, we also have half the lab working on the development of a new type of implantable stimulator that can deliver direct current stimulation safely. And so that effort has primarily been led by myself and my lab mate, Chao Jun, who is also on the call and available to answer any questions later on if you'd like. So to start, I'd like to talk a little bit about why using DC actually isn't that common, and why we're trying to explore potential applications and how to make it possible in real life. So traditional, this is an oversimplification, but traditional neurostimulators primarily consist of a metal electrode that's implanted in the body near excitable tissue, whether it be nerve or other, yes. So, and then to excite that nerve, you would deliver a negative current, which is depolarizing. So when you deliver a negative current, you transfer electrons to the metal electrode, and that negative charge buildup moves the ions in the aqueous environment around the nerve, which triggers an action potential. So this is excitatory. And then if you were to try to deliver DC using this metal electrode method implanted near the nerve, then eventually over time you would have so many electrons build up on the metal electrode that it wouldn't be able to contain them anymore, and they would start jumping to either ions or water, and you would have these irreversible, harmful electrochemical reactions that might produce like toxic byproducts or gas in the tissue, which is not good. So traditional neurostimulators use what's called charge balanced biphasic pulsatile stimulation. So basically you deliver a short excitatory pulse, and then you deliver like an equal and opposite positive pulse to pull back all the electrons off your metal electrode, so that if you deliver a bunch of these pulses over time, you kind of have no net charge accumulation on your electrode in the tissue, so you can avoid byproducts. And so while this is a really useful stimulation waveform, it's actually only really good to excite tissue. You can't really directly use it to inhibit because you always have to deliver an excitatory pulse. So that's where DC stimulation comes into play. So with DC, you can both directly excite and directly inhibit neurons. And so one example that we've been exploring in lab is so in your head. In your head, you have a vestibular system that basically encodes your head rotations and delivers signals to your eye muscles so that your eyes always move in an equal and opposite motion to stabilize your gaze. And so when you move your head in one direction, basically the firing rate of that information increases to move your eyes in a certain direction. And then when you move in the opposite direction, the firing rate decreases and your eyes would then move in the opposite direction as well. So in someone who doesn't have a functioning vestibular system, they might use a vestibular implant that encodes that firing rate to the nerve to control the eye muscles to move the head. So here on the left-hand plots, you can see on the top that you're able, with the pulsatile stimulation, you're able to encode head motion and get the appropriate eye response in one direction really well, but on the inhibitory side, you get a pretty small range that you can encode. Whereas when you use DC stimulation, you can encode really well in both directions. So that's just one exciting application that we've been looking into. And so now for the rest of the talk, I want to talk about our device specifically. So and the development that we've been working on. So the goal of our device is to create this microfluidic system that maintains a constant ionic DC output across the nerve or tissue while keeping all the electrodes within the device charge balanced, so we don't get any electrochemical reactions. So the important elements of our device are the 3 electrodes that supply current, the 2 valves that can help manipulate how the current will flow through the microfluidic system, and then the current sensing element, which is basically just a feedback sensor to monitor how much current is actually going through the output. And so On the screen, you'll see pictures of the actual microfluidic system of the device, and then up, if you can look at my little individual camera, this is the actual device in my hand. And so this is a picture of the microfluidic system, and then here's the electronic board and some like all the components that go into that. I don't really have enough time to fully talk about how we developed this board, but if you have any questions for me or Chad June later, after the TNT showcase, about any of the engineering challenges we faced or how things work, feel free to reach out and we'd be happy to talk about these things further. But so for the demonstration of how this device actually works, I'd like to walk through how we control the valves and the current supplies over time to maintain a constant DC output. So if you look here in the center of this picture of the device, stay focused on the green arrow, which represents a positive DC output. And I'm going to show you how we can control everything to maintain that as a constant DC output over time. So to start, both valves are sort of open. Valve 2 is actually starting to close. So this is kind of the first sequence of events that happened. And at this time, the first electrode is controlling the current that goes through the tissue. And ER at the bottom here is like the return electrode. And so electrode 2 is ramping down. And so on the right, you have, on the top, you have current versus time for all of the electrodes and the output. And then on the bottom, it shows you how the valves are operating in terms of impedance. So if you have a low impedance, that means the valve is open, and a high impedance means the valve is closed. So to start, yes, you have electrode 2, the current is ramping down, and electrode 1 is in control. And then once valve 2 has completely closed, then you can actually discharge all the current that's on electrode 2. So the current is now in the opposite direction and travels along this parallel side path. And not through the tissue because the valve is closed, so there's no path for the current that way. And electrode 1 is still in control. And the feedback sensor, say there is any leaking or just variation in the output, the feedback sensor is what maintains that constant output. And so then after a while, you can turn off electrode 2 and begin to open valve 2 again. And once both valves are fully open, you can turn on electrode 2 and start to transition to turn off electrode 1. So that's what's happening here. You're ramping down the current that's being supplied by electrode 1 and closing valve 1. And then it's pretty much the opposite of what happened before. So now electrode 1 can discharge while electrode 2 is in control. And then, so the same thing happens where you turn off electrode 1, open up valve 1 again, and just really quickly, you can kind of see how if you just keep cycling through these states over time, then you can maintain a constant DC output. But if you look, so this is an example of just a positive output, a MATLAB model, and then also a real device output. But in both cases, if you were to sum up like the area under the curve over this length of time, like this iteration of the, we call it a state machine, then the then the charge on every electrode sums to 0. And so, like I said, on the right, this is a recording from like the real device where we're operating the microfluidic valves again. So this is what we use to create that plot. So it's matches the MATLAB model decently well. And then we can also like deliver, this is an example of a negative output. So you can put the current in both directions to deliver excitatory or inhibitory stimuli. And so with that, I would like to thank everybody in the lab and open it up to any questions.
Great. Great job, Grace. So I'll start. I saw that there was a question from Alan Chen. Do you want to go first?
Yeah, hi. I really liked your talk, Grace. I'm wondering if you guys know like how small you'll be able to get these devices in the future.
Yeah, so that's always the first question that's asked, is that this is pretty big. So right now, the reason this is so big is because we're sort of limited to using like off-the-shelf integrated circuits. We're not trying to we're not at the stage where we can design our own integrated circuit. And so it's kind of for like rapid prototyping and being able to build things by hand. So that's why it's a little bit larger. It's not ready to be an implantable device just yet, but we're hoping pretty soon, once we have the proof of concept nailed down, to get it smaller. And so we are limited a little bit to how small we can make the microfluidics, but right now they are this size also because like they're manufactured by hand, so with a 3D printer. So we're not sure the exact limits of how small we can go, but that's why the device is the size it is right now.
Okay, but could you conceive of like a multi-channel probe, like of these kind of things going into like an area of cortex and hitting multiple like columns and stuff like that?
So we have been sort of thinking of how we could make a multi-channel device in the context of the vestibular system, just because there's multiple semicircular canals that encode for different like axes of rotation. So that's where we've kind of been thinking about doing a multi-channel device, but it's going to require quite a bit of engineering work to figure out how to miniaturize it that small so we can make them independent sources.
Okay, thank you.
Okay, so I think we have time for a little bit of judge feedback. So Chuck, do you want to go ahead?
Yeah, I guess I'm a little surprised with the size of it. What's the proximity that you have to get to your nerves is one, and the other is there's not a hardware, just an electrical way of putting electrodes in the area where if you enable and disable, you can, I guess it's depolarizing an electrode when instead of using the microfluidic model.
So when you have the electrodes directly next to the tissue, you need to, you can't really then, any charge you deliver, you need to be able to discharge that. And so if you were then to just discharge it next to the tissue, it would do the opposite effect for whatever you want. So we do kind of need to separate the interface, and that's what we use to charge balance the electrodes like apart from the tissue. And then so you were saying about the size.
Is the size, how close,
yeah, so,
if you're gonna have to be in proximity.
Yeah, so right now we actually, Chao-Jun has developed a nerve cuff that has microfluidic leads. So we actually we've been exploring like peripheral nerves and like the sciatic nerve. So it wraps around. So we have to get pretty close to the nerve. We haven't really been exploring so much in the central nervous system yet, which is a little bit trickier to navigate how we're going to place the electrodes.
Do you have a sense of how small you might make it with ideal technology?
So the problem with using ionic current and like microfluidic leads is that the smaller you go, the higher impedance that those channels are to deliver. So I think in the when we've been stimulating the vestibular system, we've been using pipette tips that are about 200 microns in diameter. But yeah, so it's kind of a trade-off that we have to investigate, like how much current can we actually output when we start getting smaller and smaller.
Yeah, okay, thanks.
Okay, thanks. So there will be time for more questions at the end, but great job, Grace. And we're going to move on to our next speaker. So up next, we have Dr. Rachel June Smith from Dr. Sri Sarma's lab. So take it away.
I have to unmute myself in order to do this. That would help. So y'all can see my slides, right?
Yeah, perfect.
Awesome, awesome. Okay, well, thank you so much for having me. My name is Rachel Smith. I'm a postdoc in Sri W. Sarma's lab in the Department of Biomedical Engineering. And I'm going to talk to you about my project today looking for resonance phenomena in the brain. And we've specifically looked at this in reference to epilepsy. So epilepsy is a neurological disease that affects about 1% of the world's population. And it's the disease of having recurrent seizures. And about 30% of these patients are what's called medically refractory, which means that they do not respond to anti-epileptic medication. And of these patients, there's about 50% that have what's called focal MRE, meaning that their seizures are coming from a specific part of the brain. And so the idea is that if we can find what part of the brain the seizures are coming from, which is called the seizure onset zone, you can take that part of the brain out, and then hopefully that patient after surgery would not have seizures anymore. So we want to find some ways to find where that seizure onset zone is. So the way that they do that right now is with intracranial monitoring. And so in order to do intracranial monitoring, they will, you'll first do typically a craniotomy where they will implant either like a grid of electrodes, or they'll implant these depth electrodes called stereoelectroencephalography into the brain. And then the patient will just sit in the epilepsy monitoring unit for anywhere from days to weeks, waiting for seizures to happen. And then the clinical team will then look at all of their seizure data and look for specific features in the data, such as high frequency oscillations or paroxysmal shifts, in order to determine where the seizures are coming from. But the problem with this is that the clinicians need a lot of seizures in order to accurately determine where the seizures are coming from. And this is a very imperfect system. Currently, about 50% of patients will still have seizures after surgery. So we need to find a better way to do this. So one way that we want to first investigate this is, you have these patients coming in and they're waiting passively in the EMU for seizures to occur, but what if there was a way that we could actively elicit the patient's seizure network? And this would decrease the length of stay in the EMU for the patients because they wouldn't have to wait so long for seizures to happen. And that would decrease the risk of infection that happens from these electrodes being implanted in their brain for so long. And it also would increase our certainty of what areas are actually causing the seizures if we know that we can actively probe this network. So what biomarker do we look for this? So I'm going to posit that that looking for resonance phenomena in the brain may help us answer this question. So there was this incident back in 1997 of a TV show in Japan called Pocket Monsters, and they in the show showed a flashing light at about 12 hertz, and about 700 kids in Japan ended up having seizure-like symptoms and showed up at the hospital. And so this kind of is an example to show that there are periodic stimuli that can induce either, if the child already had epilepsy, then induce more of these seizure-like activity that happens in their already pathological parts of their brain. But also there were some healthy kids that had seizures from this TV show, which is pretty interesting to say that there are parts of our brain that may be more resonant in the sense of you treat it with you stimulate it with a periodic stimuli, and you get this like magnitude or spread of probably pathological activity that wasn't there before. So we hypothesized that what if we looked for this in all focal epilepsy? Are there regions of the brain that are more resonant and that we can use as a proxy for being possibly more epileptogenic, because we see seizures starting in a region and then propagating to the rest of the brain? So in order to talk about this, we need to talk about resonance in physical systems in general. So the way that you can investigate resonance in a system is you can look for, basically we can think about this in terms of like an example of a child being pushed on a swing, right? If you push them at a too fast of a frequency, then they're not going to go very high, right, on the swing. But if you push them at a very low frequency, they're also not going to go very high. But if you push them at exactly the right frequency, or that natural frequency or characteristic frequency of the system, then you can get the child to swing very, very high. So this amplitude is going to be very, very high if you're providing periodic stimulus or input at the system's characteristic frequency. And so this plot that you can see on the right here is called a magnitude versus frequency plot. And the way that we can actually get this in physical systems is by looking at the impulse response of the system. And so the way that we want to look at the impulse response of the brain is by using what's called single pulse electrical stimulation. And this is data that we've gotten from the hospital where the patients have these electrodes implanted intracranially. You can stimulate a pair of those electrodes and look at responses that happen in the rest of the brain. And so from this kick, right, from this impulse, we can build a system out of these responses and then generate a magnitude versus frequency plot like the one you can see on the right. And this is showing what the magnitude of the response would be if you input a periodic frequency that's equal to this frequency on the bottom. So we hypothesized that stimulating a seizure onset region will generate really tall peaks in this magnitude versus frequency plot, and that indicates resonance, right? So in order to build these models from, and build our models from brain data, we first just look at a simple state space model that has our A transition, state transition matrix, and B our input vector. And then we're modeling U of T as just an exogenous input as a single pulse. And then, so from our state space models, we're able to turn them into transfer function models and get out our magnitude versus frequency plot. And then from these plots, we can calculate features of the Bode plot, such as that peak amplitude and the range of frequencies that the peaks occur over. And then we can calculate what we are calling a PW ratio, which is so basically it's just how peaky that the peak of the Bode plot is, right? And the peakier it is, the higher the PW ratio. So if we look at this in patient data, okay, so we're retrospectively looking at a patient who was a surgical success, meaning that the clinicians treated the electrodes that they clinically on their own determined were the seizure onset regions. And when they removed the regions of the brain that they thought were the seizure onset zone, the patient was a surgical success. So we know that the regions that they have defined as a seizure onset zone were epileptogenic. And as you can see, the PW ratios are very high, more for the SOZ regions than for the non-SOZ regions. The red is the SOZ and the gray is the non-SOZ regions. And if you look at the Bode plots on the right, you can see that the red lines are typically peakier and taller and peakier than the gray ones, indicating that there's more resonance in those regions than the non-SOZ regions. And similarly, if we look at a patient that was a surgical failure, right, that means that the patient was treated surgically and they still had seizures afterward. And then you can see that the regions that the clinicians had picked out as the seizure onset regions were not very peaky and were not very, and they didn't have very high gains. And those, so basically we think that there may be some regions of the brain that the clinicians may have missed because maybe they were, so I think that this may be able to show the clinicians that there are regions of the brain that they may be missing if they were able to have access to some of these resonance algorithms. And one other thing that we're exploring right now is the idea of triggering seizures. So sometimes these patients will come into the EMU and they'll sit in the EMU for weeks and actually never have a seizure. This actually happened just a few weeks ago. So in order for them to do localization, they need to be able for the patients to have a seizure. So there's clinical need to figure out how we can trigger seizures in these patients with epilepsy so that when we can make sure that we record them in the epilepsy monitoring unit. And this is just an example of a patient who had already done, they were actually about to get explanted the next day, and they were just doing some functional mapping, which they do at 50 hertz, and they actually stimulated a seizure in this patient at LAH 7 and 8, which is an electrode that was not recorded as the seizure onset zone region. And when they stimulated that region at 50 hertz, they got a seizure. So thank you for your time. I really appreciate it.
Great. Thank you very much, Rachel. Actually, Thea had a question. Thea, want to go ahead?
Yeah, sure. So great talk. I was curious when you're showing the different resonance peaks, sometimes they occurred at different frequencies. Do we know why they might occur at different frequencies for different electrodes?
No, and that's a great question. And we're still kind of exploring what that would mean. So it's interesting. A lot of these peaks are happening at very low frequencies, which is actually where we investigate with the single pulse electrical stimulation is actually right around, anywhere between 1 and 2 and 3 hertz. So, Yeah, so it's pretty interesting that some of these peaks occur at off frequencies, and we're still like working with clinicians to be able to do kind of these systematic tests of, can we stimulate at frequencies that are too low and frequencies that are too high, and then kind of see how either you know, the responses to electrical stimulation change in the waveform, or if we can elicit a seizure in a way that was unexpected clinically. Yeah.
Cool, thank you.
Yeah.
So I actually have a question myself. So when you say stimulation, you're talking about direct current stimulation, is that right?
Yes. So we use, so there's inside, so in the context, you can just emit a single pulse of current into the brain. Yeah.
So, and just out of curiosity, I mean, with these patients, did anybody try it with the transcranial magnetic stimulation, so you can actually target different areas of the brain and see maybe where you targeted your TDCS probe is not correct, but maybe neighboring things or out there would be a better place?
Yeah, that is not something that we have looked at all, but I think that would be so interesting. Yeah, for sure. This is so they're actually doing this more for a study. It's called cortical evoked potential. So it's kind of this area of study where you have these specific types of stimulation that you're using, and you're kind of looking at building connectivity networks based on how these different peaks look in the remote areas. But yeah, connecting that to other types of stimulation would be incredibly interesting. We have not done that.
Great. May I ask Dr. Wolstein and see if he has any input for you?
Is that me?
Yep.
Yeah, it seems cool. I mean, I think it's a really hard problem because there's so little data, right? You have to work with these surgical patients, and you don't really have the ground truth. And you know, I just think it's fundamentally hard.
A very hard problem. Yes.
So, you know, hope you get good data and lots of patients, and you can keep making progress. It seems like you are making progress. That's exciting.
Oh, thank you so much. I appreciate that.
I have a question.
Go ahead.
So you're treating the brain in this case as a linear system, right? I mean, impulse response is a linear characterization. To what degree is that premise, you know, reasonable?
Sure. That's a great question. Happening though is because you have this kind of like if you were trying to measure spontaneous activity and build a linear model that match spontaneous activity, you have to use extremely short time windows. It doesn't work very well. But since you are kicking the system with this impulse, you're actually like just doing that instead of trying to measure spontaneous activity actually works a lot better with a linear, so basically you get better output out of your linear model than you do if you're trying to measure spontaneous activity. So so this is actually a better application for a linear model than even some other things that you would do is just spontaneous EEG.
Great, thank you very much. And for the sake of time, let's move on to our next speaker. I will our next speaker is Luke Osborne from APL. And Luke, would you please take over?
We don't see anything yet for screen sharing.
Sure, give me just 1 second. Computer's having some issue. Sorry, y'all. Anything yet?
Yeah, we see it now.
Yes, we can see it.
Great. Okay. Thanks, y'all. So I'm Luke Osborne. I graduated from the VME program a couple years ago and have since moved to the Applied Physics Lab, where I work in the Research and Exploratory Development Department, which is basically their the research division of APL. So the work I do is largely dealing with prosthetic limbs or rehabilitation and things like that. So what I'm going to show here is some recent work we've done to start trying to translate our work with prosthetic limbs from the laboratory to somebody's home and how that might affect their performance. So the big question is, what value does a really expensive, fancy prosthetic limb provide? So this individual here on the right has had an amputation where most of his arm has been removed, and he has an implant coming out of the bone that we're able to attach this prosthetic arm to. And the goal of this study was to evaluate how long-term usage, so over the course of a year, long-term usage of a sophisticated prosthetic limb would affect his functionality and his behavior. And importantly, what happens if we kind of give him unconstrained use of this system? And what makes this study possible is that there's a lot of onboard data logging and wireless control that's all being handled by what's in this prosthetic limb. And we were pretty selective of who the participant was because we wanted somebody who had a lot of experience and was really going to put the system through the ringer. So this guy has had over 8 years of experience using this particular prosthetic limb. All right, very quickly. The hardware, the part of the system is the modular prosthetic limb, which is kind of shown here in this cartoon. And there's a whole bunch of position sensors and torque sensors, and we're able to connect it wirelessly to EMG, so electromyography. This measures the muscle activity on somebody's residual limb. And we're able to build this custom attachment so that it could interface with Johnny's implant. So Johnny is the participant. And basically there's a processor that's kind of stuck within this module that then physically attached to his body. So this in itself is pretty exciting because it lets him take the arm on and off very, very quickly. But that's not what we're going to talk about here. So some of the big takeaways that we started to see from this study was how his usage of the prosthetic limb changes over time. One of the goals of, say, creating a prosthetic arm is that you want to help restore functionality and its quality of life to individuals who might need them. And so some of the things that Johnny traditionally wasn't able to do as well with some of his previous devices were things like till the garden or cook or do things like try to play the piano. These were activities of daily living that he started to do more and more of as he was using the limb. But from the data that we were able to extract, one thing that we saw was that his usage of the prosthesis was slowly increasing over time. And an increase of 1% per week doesn't sound like a whole lot, but it's actually a pretty exciting thing to see, especially from a user like Johnny, who has been kind of at the forefront of prosthesis research for a long time and is well versed in different devices and systems. And so this plot here in the bottom is showing on the vertical axis how much time the prosthesis, the modular prosthetic limb, was turned on and being used over the duration of the study over this 52-week period. And if you kind of plot the trend over time, you'll see this gradual increase as the study goes on, which is exciting and reassuring because it means he's using the limb more and more. Something else that we saw is that his ability to control the prosthesis also improves. And as part of the study, he had this mobile phone interface where he was able to connect to his arm and basically decide which movements he wanted the prosthesis to be able to make, or how much training data to provide to the machine learning algorithms that were on the arm, and basically taking in those muscle signals and then converting that to prosthesis movements. So he kind of had complete control over what he wanted to do with it, and which movements to make. And what we saw is by giving him this kind of autonomy to pick and choose what he wanted, we saw that his what's called control accuracy was improving at about 0.5% per month. So here on this plot, the vertical axis is showing the accuracy of the algorithm that was being used to basically convert his muscle signals into prosthesis movements. And what you'll notice is that it's pretty good to begin with, kind of hovering in this like 90 to 95% accuracy range, which is really good for somebody who's trying to control a prosthetic limb. But as time went on, he continued to get better and better at using this system, which is also exciting because he, in a sense, is an expert and has been doing this kind of thing for the better half of a decade. But given the chance to kind of explore and use this advanced arm in an unconstrained setting, we're seeing that his improvement continues to go up. One thing that we were not expecting to see that we started to notice is that not only did his control improve, but the efficiency of that control also improved. So the top row is showing some of the common movements that he would make with his prosthesis. And these plots, which I'm about to animate, are showing the muscle activity for each electrode. If we were to take one of these electrode bands, these EMG bands off of his arm and kind of look down the center of it, and you plotted the signal amplitude, what would that look like over time? And we noticed that as the study went on, that amplitude began to decrease and go down. So basically the amplitude of that signal is decreasing, which is indicative of reduced effort, basically being slightly more efficient at making those movements with his prosthetic limb. So he's using less energy to control his device as time goes on. And the way we look at this and quantify it is by plotting the mean absolute value across all of these different electrodes and kind of normalizing it and looking at it over time. So that's what these plots on the bottom are showing. And I've highlighted a couple of the unique movements where that trend is particularly a little bit more obvious, where we see this decrease in essentially the absolute value of that muscle signal that he's producing to control the device. Okay, this is all like exciting and awesome, and as an engineer, it's really cool to build a system, put it out in the wild, and be able to collect awesome data from it. But one thing that this system now allows us to do is basically let users define their own goals and decide what it is they want to try and accomplish or what functionality they need to be successful. And so I'm going to play this video and just thumbs up if you can hear it. But this is kind of one of the reasons why we want to do this type of thing. Awesome. Absolutely. Yeah, very inspiring. So as we wrap up here, I think it's important for us to keep in mind that it can be pretty powerful and motivating for users to have these defined goals that they set to help us really evaluate long-lasting impacts. And that's kind of the motivation behind why we might want to start exploring and running these studies where we translate our technology from the laboratory and benchtop to patient's home. So with that, I'll end there and happy to take any questions.
Perfect time and really amazing work, especially the video at the end was amazing. It looks like our first question actually will be from Chuck. because he was the first to write in the chat. So go ahead.
Yeah, Luke, it's a one-year study, and I know with employment you have seasonal fluctuations. With activity, you have different activities. So then you saw 1% gain. If you were to shift that around, might that change? Did you compensate for the seasonal activities?
Yeah, it's a really good point. We didn't, but that's something that we should do and go back and look at, because there's another participant who was able to do something similar for not quite as long, but it would be interesting to see if, yeah, if in the summertime, because that came towards the end of the study, maybe he was using it more because he was outside and things like that. But yeah, it's a great point, but we didn't do that.
And another point is that with such an experienced user, is there much of a perceptual change in going from 94 to 96%? And you might see a much bigger change with untrained. By the way, it's really impressive how many dimensions you can control with that. So that aspect is really great.
Yeah, I think what's more perceptual from the user's point of view is the reliability. And I think, so it's great to see the increase in performance, but I think the participants' takeaway was it's nice to be able to pick and choose, which movements are being controlled. And so they have a little bit more oversight. And I think in that sense, that the perception of that type of control is better appreciated, more appreciated by the participant. But you're right, yeah, you might, have a bigger impact if you go from, say, like a 50% control performance up to an 80 or 90.
Thanks.
Okay, one more quick question from Gene.
Yeah, hi, Luke. So one thing that kind of comes up with prosthesis work that I've seen is, having partial performance is not, I mean, don't get me wrong, I'm all for the prosthesis work, like my gig, right? But the question I have is, like, would this person prefer something that was static, that they could easily move around and are used to controlling, right? I mean, clearly this person was motivated and interested in providing data for the science, and I'm grateful to him for that. But the question is for, given a person an amputee off the street, would they prefer a device like this, or would they prefer at this point, at the stage that it's developed?
Yeah, I think, to be honest, they would prefer whatever doesn't break, whatever form that comes in.
Reliability is a high priority.
Yeah, that tends to drive need and above all else. So even if, yeah, maybe you could use it and get good enough to the piano, if it's breaking, 25, 30% of the time, it's not good enough. So one of the things which I didn't touch on was, there were a lot of failures, technical failures along the way, and that, drives a lot of frustration. So I would say for the general population, it's all about reliability and robustness. And is it going to do what you need it to do all the time? And if you can't accomplish that, then it's kind of not worth the hassle from a lot of people's perspective. So we're getting there, right? We're looking at better control algorithms and things like that to try to get there.
Thanks.
Okay. So we have more questions, but I think we'll just have a session at the end for anyone who can stay over to field those questions from the talks. So up next, we have our own Ehsan Nejad from Dr. Reza Shadmir's lab. You're muted, 1 second. I will start the timer for you, and I can unmute you.
So I start the timer for myself too. Okay. Thank you very much. I appreciate if people can turn on their cameras just for a second. So my name is Ehsan Nejad. I'm a 6th-year PhD student from Dr. Reza Shadmir's lab. The work that I'm going to present today is a collaboration with 2 other PhD students in the lab, Paul Hage and JP. So most of the work will be a collaboration type of thing. So before I start going to the kind of science and what we did, I want to establish the concept of the internal models of the movement. So what I want you to do now is that imagine yourself that you're in a highway. Nobody's around, so everything is safe. You are on the right lane and you're driving your car, or maybe driving your bicycle if you're not driving a car. I want you to imagine yourself and put your hands on the wheel. Okay, so I can see your hands. Put your hands on the wheel. Okay, and I want to ask you to close your eyes and change your lane in 5 seconds. Okay, so after I said go, please do that. Okay, so put your hands on the wheel, close your eyes, and change your lane. 1, 2, 3, go. Okay, great. So most of you are now off the road, kind of sorry for you. And the reason for that is, as far as I know, one person was doing this. How can you move the wheel with respect to your body? I mean, that's kind of amazing. But anyway, the reason is that you were thinking the car is moving in parallel. But what actually is happening is that the car will change its angle when you rotate the wheel, meaning that you have to rotate it back to correct the angle and make it back in the same lane. So it's actually an S-shaped movement. So this is an example that your explicit understanding of the dynamic of this car is actually incorrect, but nevertheless, you are able to drive very much correctly. So your brain has an internal understanding or internal models of this movement, and it's perfect doing it perfectly. Okay, so this was a very high level type of task, and let's do another very simple task that we are actually doing in the lab. So what I want you to do now is just track this dot on the screen. Okay, so let me see your eye movements. Okay, great. So what you were doing is that you were just looking here, looking back and forth between those two things. If I ask you what you're doing, you were saying that my eyes were here and my eye landed here and then I went back. But what happened in reality is that actually your eye will land a little bit short for kind of these high amplitude saccades. And then there's always a little bit of corrective saccade as what we call it. So even for this very simple ballistic eye movement, and what happened in reality is a little bit different about what you think it is happening. And that's what we are studying in our lab, about how does these internal models being implemented in the brain and what is the neural substrate for those sort of things. So from the patient studies and also animal lesion studies, it has been established that the cerebellum is the locus, is a potential part, is actually very important for the implementation of intermodal. So this is a patient study. This patient has a cerebral degeneration, meaning the Purkinje cells in the cerebellum has died. Can you see what is wrong with his eye movements? So whenever he makes the left far saccades, the eye starts fluctuating. But and also whenever he makes the right versicles, the eye fluctuates, but it's able to kind of converge. But a healthy control subject will make a saccade a little bit short, what we call hypometric saccade, and then correct it again, a short, and then correct it. Okay, so this is what will happen if these internal models, the neural substrate that encode these internal models will be damaged. Okay. So what we did in lab is that in 2018, I started working with marmoset monkeys and building this electrophysiological lab in our lab. So our lab was kind of just doing human psychophysics, but then we started working with marmoset monkeys. We first established the protocols. So this is the first time that these monkeys were being trained for doing this type of tasks. So we established protocols to train the animals to do saccadic eye movements. We also established protocols to do the head implant surgery. And very importantly, we established protocols to target deep brain areas so we can advance our electrodes deep in the brain and record from cerebral cortex. We also, because we are working with Purkinje cells, next slide, you will see an example of Purkinje cells. We also, from ground up, wrote a Python package software with a very cute GUI. And I'm proud of this GUI that we're using to sort the neural stuff. So let's see a sample kind of data. So this is a monkey. Instead of just making left and right eye movements, the monkey is making saccades in 8 directions, and then again another 8 directions. So it's a kind of 2-step saccade task. Here for you, I'm presenting the onset of the eye movement and when the eye movement is finishing. So these Purkinje cells that we are studying in our lab have 2 types of spikes. One of them is simple spike that's more similar to other spikes in the brain. This is a raster plot, meaning each dot is representing as happening of a one spike. So when it's sparse, it means that there is a decrease in the activity. When it's dense, that's an increase in the activity. Here is the estimated firing rate. So what you can see here is that these Purkinje cells have encoding for the eye movements, but it's not obvious what the encoding is. It's very complicated. Sometimes it's bursting, pausing, some other time it's pausing. And this has been the challenge in the field to understand what these Purkinje cells are doing, what the heck of this complicated system is doing. What we added to the kind of literature, and it's kind of what we're doing, try and wrap it up and publish it, is that since we had the software and everything, now we can also sort the complex spikes of the Purkinje. So these complex spikes are complex, so they're really hard to sort, but with our things, we would be able to do that. And complex spikes are kind of sparse, but if we now look at the probability of a complex spike happening before the saccade, what it gets revealed is that this complex spike has really beautiful tuning for the direction, okay? And this is indeed what we are going to use to make sense of this simple spike activity, okay? So first of all, let's see if we can replicate the literature and see what we can see the same thing that has been reported in literature. So if I put all of, so this is a population activity. So I put all of my 138 cells that we recorded from, we put in one basket and see the lump sum total activity of all these cells. What we'll see is that there is, as a population, they are encoding the saccadic hyphen. They will burst before the onset of the saccade, and they will pause during the saccade. But what is strange is that for different amplitudes or different peak velocities of the eye movements, these cells are not changing their activity. So it looks like they are modulating for the saccade, but they don't care if whatever type of the saccade it is. It's just firing the same way for all type of saccades. But if we now use this new framework, so if you look at these saccades not in terms of the absolute coordinate systems, but in terms of a relative coordinate system, and we define this relative coordinate system based on the tuning of the complex spike. So we are saying that this direction is the saccade onset. So now we put all of these directions for all cells together. Now, if we look at the activity of the simple spike, what we were able to see is that now we will see not only the encoding for the saccade, but also for different amplitude and different peak velocities of the saccade, these cells are encoding the ongoing saccade. So these cells, with this, we can solve this issue that how these Purkinje cells are contributing to the ongoing movement. We had to use the complex spike framework to be able to see their encoding of the, not just the saccade, but also the amplitude and also peak velocity of the saccade. Okay, just to wrap up, today I start with this very simple context, our concept of what does the interior model is. We did a very fun experiment with just simple saccadic eye movement and saw that even this simple eye movement can be different than what you think it should be. When the cerebellum get damaged, all types of control of movements also get damaged, and people may have very difficulty. In our lab, we established protocols and proved that the marmoset monkeys are very promising animal models to study cerebellum and also internal models of the movement. We developed a software package to sort the cerebral neurophysiology. And lastly, our kind of science-wise part of the talk is that using the complex spike framework, we would able to, I should play this for myself, we would able to make sense of the cerebral activity in the brain. Thank you, and I would like to take your questions.
Okay, great job. I see we have a question from Paul. Do you want to go ahead?
Sure. You mentioned that it was important to sort out the complex spikes in addition to the simple spikes, and I was curious, like, what advance exactly allowed that to be possible?
Yeah, so that's a great question. Let me go back a couple of slides so you can actually see. So it's basically something named nonlinear dimensionality reduction, a technique called UMAP. So it's an advanced, it's a modification of TSNE, if you have heard of that. But using the UMAP, we were able to actually detect just a tiny differences in a waveform. and we will able to sort the complex spikes of the cerebellum. So these complex spikes are very hard to sort because their waveform can change even from one spike to the next a little bit, and that's enough for making the PCA and kind of linear algorithm kind of obsolete, but the nonlinear algorithm was able to sort them out.
Okay, and can we turn to some feedback from Joshua?
So that seems pretty cool. I'm curious, what fraction of the behavioral variance can you explain with your new model that includes complex cells as compared to what fraction can you explain without that?
Yeah, so basically without this model, we cannot explain any variability in the data. So because it looks like that these complex spikes are just doing the same thing no matter what the type of the saccade is, what the amplitude is. So it depends on what you define as a variability. If you mean peak velocity, so our model is able to define it has a linear relationship between the peak velocity and the peak firing activity here. It also has a linear relationship between the amplitude of the saccade. So in that term, it's kind of very, very good. I cannot put a number on it because it's just a correlation to show that this model can has a very good correlation in predicting those things. Did I answer your question?
Yeah, thanks.
Okay, so then with that, we're going to turn to our final speaker, Omid Zobieri from Dr. Kathleen Cullen's lab.
Hello, can you see my screen? Okay, great.
Not yet.
Hello.
Now we can.
Okay, good. Sorry. So hello, everyone. I'm Omid Zobieri, PhD student in Kathleen Cullen's lab. So, and yeah, today I'm also gonna talk about our recording from preclinical cells and how they are encoding a predictive encoding of self motion. So the vestibular system is important for gaze stabilization, spatial orientation, navigation, and also generating postural reflexes. These postural reflexes help us to maintain our balance in response to unexpected head perturbation. After a passive or externally applied perturbation, the head movement is sensed by vestibular sensors and then transmitted to the central neurons with vestibular afferents, which in turn sends compensatory motor commands to neck and limb to stabilize the head. But when the goal is to generate a similar head movement during an active or self-generated behavior, these reflexes are against the purpose. And to prevent this, the brain must somehow distinguish between these two conditions. Our lab previously looked at the neural encoding of the circuit during active versus passive head movements and found that the vestibular afferents encode both types of movements the same way. However, the responses of the central neurons and the vestibular and deep cell nuclei are suppressed during active compared to passive head movements, which suggests that the existence of a cancellation signal projecting to these cells during active head movements to cancel the afferent inputs. Our lab previously studied the mechanism of this cancellation and found that the vestibular reafference cancellation is the output of the interferon model that expects the consequences of motor command that suppresses the incoming vestibular feedback during active movements. Only in conditions where the actual sensory feedback matches what is predicted from the interferon model. Although we know the rules underlying this computation, we don't know the neural mechanism of this cancellation. And more specifically, we don't know the source of this cancellation signal. And the prevailing view is that the neural mechanism underlying this computation is cerebellum-based. The cerebellum is commonly believed to generate the interferon model that predicts the sensory consequence of self-motion and then computes the difference between the prediction and the sensory feedback. In addition, this view is anatomically supported by the fact that cerebellum closely interact with the vestibular and deep cell nuclei, and simple spikes of the cerebral prekineal cells are the main inhibitory inputs to neurons in these regions. Thus, we hypothesized the cancellation originates from the Purkinje cells. And to study this, we answered 3 questions. First was how do Purkinje cells encode sensory inputs during passive movements? Second, do Purkinje cells respond differently during active versus passive head movements? And finally, given the answer to the first 2 questions, we wanted to know whether Purkinje cells can provide cancellation signal. So first, we recorded from neurons from Purkinje cells in the entire awareness during active and passive self-motion. Since the information about head and body movement comes from the vestibular perception and perceptive feedback, for the first question, we characterized the response of the Purkinje cells during 3 passive paradigms, which tested the neural responses to vestibular only, perceptive only, and a combination of both stimulations. First, we focused on the simplest spikes of the Purkinje cells. We tested the simplest spike responses to vestibular only stimulation by rotating monkey's whole body. Here are 3 examples of Purkinje cells. Top row shows the angular head velocity, and the bottom shows the simplest spark firing rate. As you can see, the simplest spark firing rate is consistent across different trials of head movements. However, different Purkinje cells illustrate different responses. For example, the firing rate during ipsilateral head rotation was increased in cell 1 and decreased in cell 2. And in cell 3, the firing rate increased for both direction movements. Also, there were differences in the dynamics of the firing rate, as some of the firing rates might lead or lag the head velocity. However, despite these differences, we could quantitate the rate of Purkinje cell simple spike using a linear model with 3 dynamic turns. And using this approach, we quantified the sensitivity of each direction of movement separately and then identified preferred versus non-preferred direction. Then we used the results from this modeling to compare the responses in different conditions. Next, to test the responses to the neck perception input, we moved monkey's body while head was fixed in space. And on the right, I compared the perceptive sensitivity to the vestibular sensitivity of these Purkinje cells. We showed that most of these Purkinje cells respond to both sensory inputs. Interestingly, when we added the responses to vestibular and neck perceptive inputs together, which is shown by dashed red lines, we could successfully predict the response to the combination of vestibular and neck perception during passive head and body rotation. The scatter plots compares the sensitivity of responses to head and body versus the prediction from summation model, showing significant correlations. Thus, so far, I showed that Purkinje cells receive both vestibular and neck perceptive inputs linearly, add them together during passive head and body movement. Next, we want to know how these responses are compared to the response to the condition that the monkey actively generates head and body movements. Here on left, I have an example of neural activity with the same cell during passive and active head movements. As you can see, the model from the passive condition overestimated the firing rate during active conditions, suggesting attenuated response during active head movements. The bar plots also shows the comparison between 61 Purkinje cell simple spikes during passive and active head movements, which shows markedly suppressed sensitivity during active rotational head movements for both preferred and non-preferred directions. We also looked at the Purkinje cell responses during active and passive linear movement, and similarly found markedly suppressed responses during active translational head movements. Thus, our results so far showed that the single Purkinje cell simple spike responses reduced during active versus passive head movements, which is surprising if we assume that the Purkinje cells cancel inputs from the vestibular afferents during active head movements by inhibiting the vestibular and deep cell nuclei. However, each neuron and deep cell nuclei receives inputs from more than one Purkinje cell, which is about 40 to 50 Purkinje cells from ipsilateral side. This suggests that the cancellation signal to the vestibular and deep cell nuclei could be explained by convergence of the population of Purkinje cells. So first, to gain insight about the requirement for the feasible population coding, here I showed some hypothetical examples. We can show that if Purkinje cells linearly encode head velocity, the output of their population is still suppressed during active compared to passive head movements, thus cannot provide the cancellation signal. However, if we have inputs with diverse dynamics, so we can show that although a single Purkinje cell response is still suppressed during active head movements, multiple of them can be combined in a way that can provide the cancellation during only active but not passive head movements. As I showed earlier, recorded Purkinje cell simplex spikes also showed similar diverse responses. So we modeled the linear combination of the population Purkinje cells to see whether these inhibitory inputs could generate responses similar to the firing rate of RFN neurons in deep cell nuclei during all 4 passive and active paradigms. Then we evaluated the performance of each model by R-squared as a function of number of Purkinje cells. The blue line and shades are the median and 95% confidence intervals of the accepted R-squared computed based on the known variability across different RFN neurons. And the gray and line and shades also represent the average and standard deviation of the 1,000 sets of random trials from randomly selected Purkinje cells. First, when we use a small number of Purkinje cells, the performance will be very low, which suggests that single Purkinje cell firing rate is not sufficient for generating the cancellation signal. However, when we increase the number of Purkinje cells to around 40, R-squared reaches within the acceptable band. The estimated output of 40 Purkinje cells is shown superimposed on the target firing rates. The number 40 is interesting since anatomical evidence also showed that each neuron in the deep cell nuclei receives around 40 Purkinje cells. Thus, our results suggest that the internal model that can account for the suppression of visceral input during active head movement could be encoded by population of Purkinje cells. So I showed that the Purkinje cell simplex spike activity during passive head movement linearly integrates vestibular and proprioceptive inputs. And the responses modulation is markedly reduced during active head movements. Although the reduction in single Purkinje cell is surprising, we showed that the population of Purkinje cell could still encode the internal model that expects the consequences of voluntary safe motion. Okay, Dan, thank you, everyone.
Great. Thank you very much, Omi. That was an interesting talk. Is there any question from the audience? Okay, looks like there's no question from the audience. I would, Jean, do you want to give inputs to the Omi?
Sure, I have a question. Does it, so those Purkinje cells, do they are they very specific to this type of motion, or do they respond to any other kind of motion? How specific are these?
So these Purkinje cells respond to, so we test for rotational head movements. We only tested the yaw direction, but also we tested translational in the horizontal plane, well, forward and lateral. The majority of them also respond to the translational head movements in the horizontal plane.
Interesting. So that means that if you move at all, it causes this inhibition signal. It's not just so if you voluntarily move in one direction, it inhibits everything, the other directions as well.
Yeah. So if you're talking about the cancellation of voluntary movements, yes. So any voluntary movements needs to be canceled that are being canceled.
So the trigger can come from anywhere, basically, is what it sounds like.
I think the point that we think it's true is like it happens only when there is a match between what is predicted and what is you get, and that intermodal. So like what you get, so you should have a like specific intermodal for that specific input.
Okay, thank you.
Thank you very much. And so because we're past over time and there is no other questions from the audience, I would like to pass it to Thea. So, and please don't leave because we are going to have a vote for the favorite audience. Okay, go ahead, Thea.
Sure, 1 second. All right, so hopefully you're seeing this. Great. So that's a great end to day 2. Thank you to all of our presenters from day 1 and day 2 for sharing all their different amazing presentations. So like Esan said, it's now time for you to stay to the end and vote for your favorite presentation from day 2, who's going to win a separate prize from our winner from day 1. And like I mentioned, because the competition's close, we'll not know the final voting on all the other prizes until the judges get to meet and maybe can announce it on Friday. So one more time, just to build the suspense, I wanted to also remind you guys that there are other TNT events that occur regularly. So our next one, which we hope that you guys would be excited to attend, is a neuroethics panel, where we're going to have 3 different speakers who have PhDs in subjects related to neuroethics coming in to talk to us about their work with patients, clinicians, and engineers, and trying to understand the ethical issues around designing neural implants. So if you're interested in those things, please look out for our announcement for May 20th, 2 to 4 p.m. EST, when we'll be having this panel. And you can bring your questions from your work. They'd be excited to talk to you. So with that, please follow us on social media to hear about that, our academic talks and other events like this. And we can now turn to the voting. So I'm now going to give you guys all a poll, and please let us know what you thought of today's talks and vote for your favorite out of all of them. And we have all of our votes. So it looks like we just have, wow. We have a tie. Okay, I think we have a tie between Rachel June Smith and Grace Foxworthy. And so I think that we'll take all of these votes in, and I think we'll be giving out even more prizes than we expected. And we'll let you know when the final prizes are announced, if any of you guys are multi-winners and what's going to happen. So congratulations again to all of our speakers, and thank you everyone for attending. And hopefully this is the first of multiple showcases, and we'll see you next year.